On behalf of whole ISFCP family, I, Dr. Pooja Chawla, Professor at ISF College of Pharmacy, welcome you all on the third and final day of three-day international conference, that is ISFCON 2021, Recent Advances in Pharmaceutical Sciences, Vision 2030. Today, the speakers, we have five technical lectures by the learned researchers across the country. The first one being Dr. Gaurav Goyal. He is Head Technology Transfer at Shilpa Medicare Limited, Bangalore. And the chairperson for this technical session is Dr. Sushil Kashyap, Associate Professor, Dr. H.S. God University, Sagar. I welcome Dr. Gaurav as well as Dr. Sushil Kashyap. Very good morning to both of you, sir. The subsequent lectures for the day would be delivered by Dr. Amit Agarwal from PGI Chandigarh, Dr. Jayant Karajgi from Shipla Medicare Bangalore, Dr. Jayagopal Mehar from Dr. Reddy's Labs, and Mr. Ranjit Bhashikar, who is CEO QBD International. So I welcome our first speaker of the day, Dr. Gaurav Goyal, and now I hand over the mic to Dr. Sunil Kashyap, who will introduce you with our first speaker of the day, Dr. Gaurav Goyal. Over to you, sir.
Thank you, Pooja ma'am. I welcome everyone and Gaurav Goyal sir. So we are going to start our first session, and for this I'm thankful to everyone that they have given me this opportunity. So good morning, everyone. At the outset, I would like to pay my sincere thanks and gratitude to management, organizing team of the international conference, and my senior and mentor, who is director and principal of ISF College of Pharmacy, Moga, Professor Gupta sir, for giving me an opportunity to be part of this prestigious occasion. ISF College of Pharmacy has gathered the galaxy of stalwart in this conference for the benefit of students and delegates. I congratulate the team for the successful organization of this mega event. I am also privileged, or rather obliged, to read the CV of one of the greatest scientists working in our country, Dr. Gaurav Goyal sir. So in brief, I will read the CV of sir for the benefit of all the listeners. Dr. Gaurav Goyal sir is currently working as the head of Technology Transfer Department at Shilpa Medicare Limited, Bangalore. He has total 13 years of industrial and academic research experience. He served as a professor and head of the Department of Pharmaceutics at ICF College, Punjab, and worked with renowned industries such as AIIMS Drugs and Pharmaceuticals and Ruzen Pharma Limited. Dr. Goyal has completed his doctorate and master's studies in pharmaceutics from one of the renowned institutes and my favorite institute, ICT, which was earlier called as UDCT, Mumbai, and Bachelor of Pharmacy course from Jamia Honb'le, New Delhi. Dr. Goyal is first Indian scientist. We are proud of Goyal sir. Dr. Goyal sir is first Indian scientist to launch indigenously, maybe जिसको हम आत्मनिर्भर भारत के अंदर भी ले सकते हैं, launched indigenously developed generic fentanyl transdermal patch in year 2009 in association with Ruzen Pharma Limited. Its focus research area are development of oral disintegration film, transdermal therapeutic system, implants, nasal sprays, lyophilized injectables, sustained release topical and oral solid formulation. And Dr. Goyal has co-guided more than 13 Masters of Pharmacy students and delivered more than 50 guest lectures at various pharmacy colleges and institutions. He has received many awards, such as Outstanding Performance Award by Ruzen Pharma Limited for successful launch of fentanyl transdermal patches, award at 60th IPC 2008 held at New Delhi for the poster entitled Symbiosis Between Global Warming and Pharmaceutical Industries. Then Best Poster Award at 12th APTI Convention in 2007 held at Chandigarh for the poster entitled Anti-Counterfeiting Solutions to Pharmaceutical Industry. IRF International Travel Grant from IPA to attend 35th CRS annual meeting in 2008 held at New York, and JRF Fellowship by UGC. He is life members of many associations such as IPA and AAPS USA. He worked actively as a part of scientific committee of IPC, uh, which we call as Indian Pharmaceutical Congress Association. Brief introduction about the sir. I request Dr. Goel to deliver his talk on regulatory requirement of new dosage forms. Now over to you, Goel sir.
Thank you very much, Dr. Sushil Kasher, for such a brilliant introduction. Thank you very much. I thanks the management of the ISF College of Pharmacy to invite me as a resource person in ISFCON to 2021 conference, international conference on recent advances in pharmaceutical sciences vision 2030. This is the third validatory day of the conference. I start this particular talk with some motivational words that in this pandemic period we can think of that India where we as a placement India across the world where we are. So we can see that in across the world there are only 6 vaccines which are approved worldwide, and one of the 6 vaccines, that is Covaxin, is the indigenously made vaccine in India. So we should respect the scientists of India, and we should be very proud of India that we have such technologies where we can develop as well as market such type of formulation which are very complex type of formulations in India. So salute to the scientists of India. We are the largest manufacturer and exporter of vaccines. That is also one of the proud moment that we are not only supplying to giving to the Indians, we are supplying across the world also the vaccines. That is a requirement of the worldwide. So this is and the third one, which is very important to understand by all of us, that India is the largest number of FDA approved plants, and we are giving 40% of the American medicines, US medicines from India, that we are supplying the Indian medicines from India, the US population. So these are some of the very important points which we need to understand, that where we are placed. So with such motivational words, I start with the presentation. Okay. Right. So I think my presentation is visible to everybody.
No, sir.
Yeah, yes, sir, it is visible.
Yes, sir, it's visible, sir.
Visible.
Yes, it is there.
Okay. So I start with that.
Yes.
With my presentation. The presentation entitled is Regulatory Requirements for New Dosage Forms. And this presentation is basically for the teachers, students, and covers my scope of making this presentation and presenting this presentation before you, right? And at the end, there will be question and answer session. You can ask the questions to me, whatever there is a understanding difficulty between this presentation. Slightly, maybe I am fast at certain points, 2 or 3 formulations, where a regulatory requirement of 2 or 3 formulations. So you can ask the question later after the presentation, right? Just a minute. Yeah. To the outlook of the presentation, why we require the regulatory guidances from the regulatory agencies across the world, what are CTD modules, and in this my presentation, I will cover about the product development and regulatory concentrations for 3 of the major dosage forms, which are new dosage forms, transdermal patches, long-acting injectables, and nanotechnology products, in which I will cover basically on liposomes. So why there is a need for the regulatory guidelines? Because if we want to market something into any of the countries across the world, like US, Europe, Australia, South Africa, so there are various agencies across the world which gives us the guidelines how we can market our product into their respective countries. So these guidelines will be very helpful for them. So today I will cover only the common guidelines which are across the world, which are given, and those guidelines are through ISH. So International Conference on Harmonization, which gives those guidelines, and then guidance document for dossier filing, how we can file the dossier into the respective countries, what are the expectations of the regulatory agencies for the filing of the product, what is eCTD filing, and harmonization for filing of the products worldwide. So this is the how we can do the eCTD. Now there is no paperwork. Basically, you can file your dossier, your things through eCTD. That is common technical document CTD. Through eCTD you can find out, and this is the harmonization across the worldwide, and that is why we require to understand the regulatory guidance, and we should develop our products which in the laboratories according to those guidelines, so that it will be easy for us to immediately apply for the license, right? What are the types of drug applications which we need to after our do those studies as per the guidelines? One is the IND, that is the investigation new drug application. This is for the clinical studies. So once our product is stable, we manufacture or develop our products, we do some preclinical studies in animals, and when we prove that it is safe into the animals, it does not have any toxicity, we apply for the IND, that is investigation new drug application, and then we do some clinical studies like phase 1, phase 2, phase 3, and phase 4 is the post-marketing approval. So after phase 3, you can market your product, and then phase 4 study starts. NDA is the new drug application, which is entirely, if it is a new drug, nobody in the world is having such type of medication or dosage form, or it is a new drug itself, new API itself. So it comes under the new drug application. AND is the generic mode of application, whereas we like apply for the generic if we are making some copy of the previous innovator product, so it goes through the ANDA. And then the biological license application, where the biologicals are there. So in this, I want to say it is the liposomes come under this category. So I will make you to understand through my presentation what is the difference between the AND application, NDA application, and the biological license application. So if we see the NDA, the new drug application requirements, and the abbreviated new drug application requirement, that is the generic mode. So here we can see the 8 studies: the labeling, pharmacological, toxicology, chemistry, manufacturing controls, microbiology, inspection and testing. These are the both requirement for both the products, right? So we need to do all these studies. We have to understand the chemistry. We have to what are the manufacturing controls, what are the quality controls, what is the microbiology, what they should be the microbial limit test. We need to do that. We are controlling the microorganisms into the formulation by adding some preservatives or by some process control. Then what are the inspection parameters of that? So only the difference between the two when we are going with the new drug application and the generic application is the In the new drug application, we have to do the extensive studies like animal studies, clinical studies, bioavailability, which almost take around 6 to 8 years. I will show you. But in the ANDA, when we are going with the generic type of formulation, only bioequivalence will be required. So you have to show that your formulation, a test product, is bioequivalent to the reference product. And then this ANDA can be launched, product can be launched within 1 or 2 years, but NDA requires approximately 6 to 8 years. So this is the NDA mode of application, where we can see that here the 1 to 2 years is the laboratory and animal studies. Then in the 3rd and 4th year, we are doing the phase 1 studies after filing the IND. Then phase 2 studies, where we are getting effectiveness. In the phase 1, we are checking the safety of the medicine, and it is given to the human volunteers, not to the patient. But in the phase 2, we are doing the studies on the patients, and then phase 3 studies, which will be on 1,000 to 3,000 patients, and it is almost 6 to 8 years. On the 6 to 8 years duration, these type of studies are generally taken now. So if you can see, our journey starts from here, from the preclinical phase, and after the 8 years, we reach to the phase 3. So after 8 years, we generally launch our medicines, and only 27% of the people reach it in the phase 3. Most of the medicines are rejected in the 70, that is phase 1 studies. So this is the type of expenditure which is there with the new drug application processes. Coming to the common technical documents, what are the requirements for the dossier filing? This is a very common thing in which we teach to the students also. But slightly, I want to highlight here once again this particular thing. There are 5 modules in the common technical documents, where the module 1 is the only administrative document, and it is not a technical document. Whereas module 2, module 3, module 4, and module 5 are the technical documents. Therefore, it is known as the common technical document. And in the module 2, it is a summary of all the studies which we performed in module 3, module 4, and module 5. So it is a summary so that the regulator agency can understand in a very brief what type of studies we have carried out. And in module 3, it is a quality document, which is very important, like all the process validations, the analytical method validations, and how you are manufacturing BMR, batch manufacturing records, all comes under the quality. How you have developed your product, that is product development report, comes under the quality module. Then we have the module 4 and the module 5, where in the module 4, we give the reports of the non-clinical studies, that is the studies which we did on animals, and the clinical studies which we did on human beings, that is the phase 1, 2, 3 trials. So after all the studies, when we are going with the ANDA mode, the clinical studies will contain bioequivalent studies, right? Non-clinical studies, maybe you can skip for the ANDA products if it is not required. But certain times the regulatory agencies can ask you, before giving the IND permission for the clinical studies, they can ask you the toxicity profile in animals. So here is a big scope for the pharmacology department, because if they have the approval from the regulatory agencies, CDCO, so they can do such type of studies in colleges also, and many colleges are doing this non-clinical study reports also. In our India, the regulatory authority is Central Drug Standard Control Organization, that is CDSCO, and the head of that is the Drug Controller General of India, and through that it is controlling the state drug regulatory authorities. Each state of India has this state drug regulatory authority, which in brief it is known as FDA. So this is the way, this is our organization structure of regulatory authority in India. Now we come to the technical part of the presentation, that is product development and what are the quality considerations for the transdermal patches. Transdermal patches, as this conference is on 2030 and it is a new drug dosage forms, so we are doing a lot of dosage forms like tablet, capsules, syrups, suspensions, nasal sprays, and all those formulations in India, which is a requirement of India. But still, as like I can say, it is 2030, we are progressing, and So this is the presentation where I'm only covering the new drug dosage forms. What are the regulatory requirements across the world? Because we have to cope up, we have to come up with the speed and develop such type of formulations also in India. Right. So there are 4 types of transdermal patches at a whole. Matrix type of patches, reservoir type of patches, drug-in adhesive type of patches, and multilaminate type of patches. What are the difference between the 4 patches? I can start with the drug-in adhesive type of patches. Each transdermal patch has a drug layer, which is shown by dots here and green dots here. And then we have blue color, that is that is a backing membrane which is there above the drug layer, and there is a pink the lowermost layer is the pink layer, which is known as the release liner. So like you are applying the bandage, you are removing liner from the system to apply on the skin. So it is a similar type of bandage type of system where transdermal means the drug will be penetrate through your skin and go into the systemic circulation, into the blood to show its effect, to show its pharmacological effect. So it is a very simple type of system where you have the only single layer of matrix where drug is incorporated into the adhesive. It is a pressure sensitive adhesive. So drug is incorporated into the adhesive itself, and you are removing the liner and applying this patch on the skin. Whereas the reservoir contains a formulation into the liquid solvent, like it can be solvent like anheptane, ethyl alcohol, any solvent which will be there. And it is, because it is a solvent, it is liquid, it is protected by the drug controlling membrane, which is here it is shown by the purple color. After that, there will be adhesive layer, which will be useful to stick the or adhese the patch on the skin. So this is the reservoir type of system. And in the matrix type of system, we have a drug layer, but it is surrounded by it is in this the drug is not there into the adhesive. Adhesive is on the circumference. That is because if the drug is not miscible or not compatible with the adhesive layer, so we are giving in the center there will be the drug layer, and in the periphery it is the adhesive layer, which will be helpful for the sticking of the patch upon the skin. And in the multilamellar, it is the 2 or 3 layers of drug to give a very sustained release of drug for 7 to 8 days, 7 to 14 days also. So drug in adhesive, it is generally for 2 to 3 days, and multilamellar, it is for 7 to 8 days. So you have a rate controlling membrane between the 2 drug layers, so that the first lower layer will give the immediate effect, and the upper layer, which is above the rate controlling membrane, will give you the effect later on. So we have to understand one very basic thing from the transdermal perspective. Like if we are developing some generic formulation of the tablets, it is only if it is 25 milligram of any formulation, like if you can say pantoprazole tablet 40 milligram. So if we are developing the generic version, we have to develop the pantoprazole 40 milligram tablet. So we have to use 40 milligram in each tablet. But in the fentanyl, it is not the fentanyl. In the transdermal patches, this is not the case. I have given you the example of fentanyl transdermal patch. The patch which was there in the market of Janssen Select by the name Duragesic D-Trans, it is having the 4.2 milligram per patch, the concentration of fentanyl. This is for which strength? The strength of the transdermal patches is generally expressed in the hours. So it is a patch 25 microgram per hour for 72 hours. That is a 3-day patch. 72 hours means 3 days. So it is per hour, it will release 25 microgram drug into your systemic circulation. So this much gram will be permeate through your skin inside the blood. So for that, they are using 4.2 milligram per patch. But the patch which we developed in Rusan Pharma Limited, which I have launched in 2009, it is having 3 milligram per patch. That means our drug is having the more flux, so that to get with the lower concentration also per patch, we are getting this much flux, 25 microgram per hour for 72 hours. So it depends upon the selection of the adhesives. which adhesive we are using it, so that we can get in the lower concentration also, we can achieve that particular flux, that is 25 microgram per hour. And here it is in generic, in transdermal patches, it is very easy. If you increase double the area of the patch, it is a small patch. Suppose it is the area is 5 centimeters square. So if you increase the area to 10 centimeters square, it concentration becomes double. So you need not to develop the 4 formulations of all the 4 different strengths. You can develop one strength and all the other strengths will be depending on the area, because we are applying on the skin. So it is the area proportionate formulations. So that is the best part with the transdermal drug delivery systems. If we see what is the difference between the two things, between the tablets and other formulation, conventional release formulations and the transdermal patches, in the generic formulations, we are doing the generic formulation development using the dissolution apparatus. But this type in transdermal patches, we have to use the diffusion cells for the skin permeation studies. So diffusion cells, which we are going to use, that is what I want to say. It is look like this. It is a French diffusion cell where we apply some membrane and the patch over there. So above we apply the patch, it is a donor compartment. Then we have the skin layer. Skin layer means human skin, human cadaver skin of the dead body of the human. We apply that skin. Generally, during the research in colleges, we can use that animal skins also. And now there is an artificial skin which is also coming. So here we need to apply membrane, and above the membrane we have to apply the patch, so that the drug will be permeated through the skin inside the receiver compartment. Below is the receiver compartment, and we are doing the sampling from the sampling port. So if we are matching the flux here with the generic and between the reference product and our test product, able to say that it can be bioequivalent. So before going for the bioequivalent studies, we have to match this F2 value here, that is the similarity factor. Now we have to understand what are the QTPP. What is QTPP? It is the quality target product profile. What we want in our product, how it should be, what should be the qualities into that particular product. So in the transdermal product, the first QTPP is that in vivo delivery of active ingredient to achieve therapeutic effect. That means our formulation having the same in vivo delivery, that when we apply on the human skin or the human beings, the flux across the skin should be same as the reference product. The minimization of the residual drug. What I mean to say here, if minimization of the residual drug means here, because certain times like fentanyl is a very important case, because fentanyl is a narcotic drug. It is drug of abuse, because if it is the like the example which I just take you to the previous slide again. Here, if you see here, between Janssen Silag and Rusan Pharma Limited patches, 25 microgram per hour for 72 hours. So if you multiply this into 72 hours, it will come into the 1.8 milligram. So in 72 hours, only 1.8 milligram is permeating across the skin. But the patch contains 4.2 milligram of the drug. So that means how much is the residual amount of the drug which is left into the patch? It is almost 2.4 milligram. But in the Rusan Pharma, the same 1.8 milligram of the drug which is permeated, after the delivery of 72 hours for the 3 days, how much drug is left inside the patch? It is only 1.2 milligram. So that is half of the drug which is left inside the residual amount of the drug which is left inside the patch. So this Rusan Pharma, if we after 72 hours, if somebody will take this patch and abuse that particular patch, it will get the 2.4 milligram of the drug as a residual inside the patch, whereas with the Rusan Pharma it is only 1.2. So the residual amount of the drug is less inside the patch. That is, as per the regulatory norms, it is more better patch, and it will be getting the easy approval into the regulatory market. Then the adherence for the duration of wear period. It is very important that your patch should be stick for the 3 days. It should not be like after 1 day, if you are after 1 day it will be removed from your skin after getting bath, So it should have the sufficient strength. The adhesive should have the sufficient strength that it should remain on your skin. Then it should be minimization of the irritation. It should not be irritant. The excipients which we are using it should not be having the irritation potential. So to judge those irritation potential, generally these are the we are doing some irritation studies. I will tell you in my presentation. Then chemical and physical stability for the shelf life. That means the impurity should not be generated much. It should be chemical. The assay of the particular drug should remain within the limit, and the related substances, that is impurity, should remain within the limit. Right. So if we go to the sequence means critical quality attributes. What we are going to measure into our patch when we developed it. And we have the finished formulation. Now, what test we are going to do on those finished formulations? What test to get it cleared for the market? The first is the uniformity of dosage for units. That is, we have to test on it 10 transdermal patches separately, one patch, and it should be uniform of the dosage unit. This test you can refer from the pharmacopoeia. Assay is one of the very important tests. Permeation enhancer content. This is a very important test. We are not doing the assay of only API, active pharmaceutical ingredient. We are parallelly doing the assay of permeation enhancer. If we are using some permeation enhancer, then it also needs to be evaluated during the shelf life, during the stability studies. Impurities, in vitro drug release profile. This is very important, which I want to make you to understand. During the development, we are doing the development studies through drug release. That is the drug permeation. It should be matching with the innovator reference product, right? That is during the drug development. But during the daily manufacturing, routine manufacturing into the industry, then when we are not using this French diffusion cell, we are not doing the drug permeation and again. And how we are going to do it? We are going to do through in vitro drug release profile. In vitro drug release profile means similar to dissolution, in which we are using USP5 and USP6 separators. How these separators look like, I will show you in my next slide. Then preservative and antioxidant content. If it is a preservative, then we have to do the preservative efficacy testing, and the antioxidant efficacy testing should be done into the product, finished product. Peel addition, tackiness. Peel addition means how much force we will require to remove the patch from the skin. Tackiness is how easily we can apply the patch with the slight amount of certain things we apply with the slight pressure and it stick to the skin. That is known as the tackiness. Though peel addition should be more, tackiness should be less. With this small pressure, we can able to stick that particular patch upon this skin. Then we have release liner peel strength, shear strength. Shear strength is how the patch, when we apply on the skin, it should not be moved from one place to the another place. It should not be. The adhesive should be such that it should remain on the one place on your skin. It should not be moved from one place to another by slight pressure. Like if you by the thumb you are applying some pressure and it will move from your skin from one place to the another. It should not happen like this. Cold flow is one of the important parameter which we have to see. Cold flow is the phenomenon when we apply, we are keeping the patches. Certain times the adhesive get out of the patch, and it will come as the adhesive on the boundaries of the patch. So that type of phenomenon should not be there. Residual solvent should be controlled because we are making these patches with the organic solvents. So residual solvent content should be in the control. Residual monomers content should remain in the control because monomers are small structures which can go inside permeate through the skin. So the residual monomer content from which the polymer is made, so we have to control the monomer concentration inside the polymer. So when we are purchasing some polymer or we are using some polymer, then we should know how much is the residual monomer inside that. So my residual monomer concentration has to be controlled in the transdermal drug delivery systems. And the last is the microbial limits, which is very important. So this is a peel addition tester. I want to show you some video how this peel addition, how to this in the industries we are applying this particular patches on the SS plate. and how much force it is required to remove the patch from the steel plate. So see here, this patch we have applied, and it is 180-degree peel adhesion tester. It will be now this will be removed, and this force will be measured. See here, it is removing 180-degree up towards up. It is, and the force which is required in the load cell, it is measuring that particular force. So this is the peel adhesion tester. Now, what is shear adhesion? Shear adhesion, as I told you, it is the force which is shear adhesion should be such that the patch should not be moved from one place to the another. So here, what we are applying the patch on this stainless steel bar, and we are keeping the load on that thing. How much force it is required to remove that particular patch from one place to the another. So it should be, if it is very high load will be required, that means it is a good patch. So we can see through the video. The videos I have made it. Now he is applying the 2-kilogram bar load on the patch. The patch is already applied on this stainless steel. It is not removing from this easily. It will slightly, slowly over the period of time, it will be removed, and that time period we are going to measure it. After how much time it will be removed. So this is the machine, the overall view of the machine, and this is the close-up. And here I want to show that we are measuring the time in minutes. How much time? So it will be 10 units can be applied simultaneously, and we are measuring the time, how much time it will be required to fall the unit from the stainless steel bar to the down. Now, what are the regulatory requirements for basic regulation? This is about the development part. Now, with that development part, certain other regulatory requirements are there for the transdermal patch. That is the BE studies, skin irritation studies, skin sensitization studies, extractable and leachables, and product adhesion characteristics. So product adhesion characteristics I have already shown you through the peel adhesion tester. And now that is we are doing it in vitro. But that product adhesion characteristics we have to show in vivo, that is on the human skin also. So that is, these are the 5 different studies which we have to do before getting our product registered into the country. My voice and presentation is okay? Everybody is able to listen, madam?
Yes, sir.
Yeah.
Yes, sir. Go ahead.
Yeah, thank you. Then we have the combined skin irritation and the sensitization studies, in which we applying the patch. This type of studies are generally done on the 200 volunteers. That is, so much more volunteers will be required for registration of the product. And it is basically the multi-center studies, which is evaluator blinded. The evaluator does not know which is the reference product and the test product. We would require both the test product and the reference product for this. It is a randomized studies to support their comparative evaluation of the skin irritation and sensitization. Sensitization, I think everybody know, or if I can tell you, sensitization means it is like vaccines. Vaccines also, it is a sensitivity test for some vaccines. Small amount of penicillin is injected inside the skin. We have to see whether it is giving you, the first dose does not give you much irritation, but if the second dose, slight second dose can give you the lot of side effects or toxic effects also. So that means it is having the sensitivity towards your skin membrane or your skin or your body. So similar type of transdermal patches, we are applying the patches on the human skin for 14 days. And after 14 to 7, it is an induction phase first. That is a 21-day induction phase, which is then we are giving it 14 to 17 days rest period. And then we are applying again the patch on the human skin. That is known as the challenge phase. If during the challenge phase, if we are getting a lot of irritation, lot of redness, that means this particular patch is having the lot of sensitization potential. So those type of patches are generally not approved into the US market or across the world. So the patch should have the low sensitization potential. So first of all, we are applying the 21 days induction phase. Like if it is a single day patch, we are removing the patch after each day and applying the new patch. And for 21 days, we are applying the patch again and again. And then after 21 days, we stop the this particular studies, and then give the 14 to 17 days rest period, and then we do come up with the challenge phase, and then we apply the patch for 1 day only. And if there is lot of redness, because the antibodies if developed inside the our body with respect to that particular medication or polymers, drug adhesives, then it will show the sensitization. So these type of studies data is required for the in the US to file that. And this we are using healthy males and non-pregnant females. Irritation score, the same, it is a combined studies. So irritation, we can see the redness. If there is no evidence of irritation, it is a score is 0. And if it is a very strong reaction spreading beyond the application site, it is 7. So 7 is the maximum score. So 0 is the minimum score. So we, the physicians are giving this score on the basis with the respect of the reference product. And if there is a edema, 4 score, arrhythmia accompanied with the edema, then 5 score. So these are the rating, how we need to give the scores for the irritation. This is very important thing, extractable and leachables. I want in India now, previously these studies were not a requirement, but now CDCO has also made this requirement from the last 10 years, I can say. So this is very big requirement in the market who knows to do extractable and leachable studies. So now we have analytical departments in the colleges. So this type of studies we can teach in analytical students who are there in the MPharm analysis, pharmaceutical analysis. They can as a project they can take this particular thing as extractable and leachables. What are the basic difference between the extractable and leachables? I can just tell you in brief. Extractables are the organic material or organic solvents or which we can extract from the packaging material, not from the drug product, from the packaging material, like from the material in which we are going to pack. So because these organic solvents, why we are doing the extractable studies? Because when we do the packaging of tablets, suppose we are doing the packaging of the tablet in aluminium, blister, and for transdermal patches, we are using some aluminium, some foils, some membranes like release liner, backing membrane. These are the things which I have shown you. So these can contain some organic compounds which can move from your packaging material inside to the drug substance, onto your drug matrix. So those extractables, when we apply the patch, those organic material can along with your drug can permeate inside your skin. So extractables are generally done on the packaging materials or the materials which are primarily in touch with your drug. Leachables is how much after like extractables is, if extractables are there, how much extractables are leached into the drug product. So leachables are done on the drug product. How much is the organic product which is permeated from your packaging material inside the drug product. It can happen, most of the times it happens like there are extractables in the packaging material, but it does not leach inside the drug product because drug product does not have the solubility for those particular extractables. If it is a water phase system, if your injection is there and it is completely water based, so those organic compounds will not leach into the water phase because it does not have the affinity for the water. So still, if you have a lot of extractables, but leachables are negligible, so still your drug product can be approved into the market if you prove that there is no leachable inside your drug product. I think I try to make you to understand this. You can go through the guidelines and certain papers, and it will be easy for you to understand more. Product adhesion characteristics. If we apply the patch on the skin, if it is, we are giving the scoring. If it is 0, that means there is no lift off from the skin. It is completely adhered. All the corners of the patch is not removed from the skin. during the period of application. If it is a 1-day patch, 3 days patch, after 3 days also, it is 90% adhered to the skin. That is, it is the best. If 70 to 90%, it is 1 score. 50 to 75, 2 score. So 0% completely detached. After 1 hour or 2 hours, completely detached. That means the 4 score. So if it is 0 score, it is the best in the product adherence characteristics. Now, this is up to the transdermal patches, the regulatory requirement of the transdermal patches, which I have covered. And now I come up with my next topic, long-acting injectables and implants. This is very interesting topic because now injectables are there, which is we apply the injections. And what are the regulatory requirement for the injectables and implants? So this is very important and interesting topic, and which we can do development of the transdermal patches. I can think it is an expensive technology that generally require the collaboration between the industry and the academia. Like I developed the fentanyl transdermal patch during my academic period in when I was in ISF, but it was in collaboration with the industry, so that it was easy for me. Now I come up with the long-acting injectables and implants, where I can say it is a biodegradable injectable microspheres and in situ implants are the basic things which are there. I want to show you here how the pharmacopoeias have with the different terminologies of the same thing, they have given us a different terminology. The first line is the PHUR, European Pharmacopoeia, that they have classified the long-acting injectables into 2 categories. One is the implants and one is the gels for injections. If you open the British Pharmacopoeia and PHUR, that is European Pharmacopoeia, these 2 terminologies you will be able to get from the pharmacopoeias. One is the implants and one is the gels for injections. So these are the long-acting injectables. In US, we have implants and suspensions, and in the Japan, we have implants and the prolonged release injections. These are the terminologies for the same type of products. So it is a difference of terminology which I want to show you, that different countries have the different regulatory guidelines, give the same type of products with the different names, right? So what is the difference between the two? Because it is very difficult for the long-acting injectables to do bioequivalency. It is very difficult to prove because our enzymes and the enzymes which are present in the other human beings are different. So generally it is because of the degradation of the polymer. So this rate of degradation of the polymer is certain times different from individual to individual. So it is very difficult to prove the bioequivalency between the 2 formulations. And then you require a lot of human beings, a lot of volunteers for the same. So it is a regulatory requirement. If you prove the in vitro, in vivo correlation, and if you develop some method where you can prove that in vitro and in vivo correlation between the 2 formulations, then it will be easy for the to launch this particular product into the market. So what is QTPP? What is our target product profile? How we want our product to be launched into the market? It should be, first of all, it should be sterile, because we are going to put that particular injectable inside the skin. It should be biocompatible, non-immunogenic. It should not show immunogenicity, negligible local adverse effects, extended drug release, like it is a long-acting injectables for 30 days. Certain times, like I have developed a naltrexone implant with the Rusan Pharma Limited, that is for 6 months. If you implant the particular, it is a tablet type of structure that we are going to put by the operation, that requires the operation. So you have to incise the skin, then you have to keep the tablet beneath your skin into the subcutaneous layer, and then you have to stitch the skin again. So this is the type of formulation. So it is for 6 months. Naltrexone, it is basically for the alcohol de-addiction. Patients who want to come up with the alcohol de-addiction, so that particular patient can use that, right? So, but the very important thing, it should be biodegradable. It should not remain inside the skin. That is very important thing inside the. And if possibly we can interrupt the therapy, like implants, there is a tablet for 6 months, but if you are getting the side effects after 5 days, so you can open that particular stitch, that area where you have put that tablet, and can withdraw that particular tablet. So it should have the possibility to interact with therapy. So these are not the two very important parameters. So these are shown by the dotted lines. But if they have this type of hello. Is it okay?
Yes, sir.
Yes, sir.
Please go ahead.
Yes, sir. You're audible.
Okay. Yeah. In there, I have shown you the in vitro in vivo correlation of the naltrexone loaded microspheres. Microsphere is a very important formulation for the extended release injectables, and the polymer which is used here, which is approved across the worldwide, is the PLGA. PLGA polymer, it is extensively used. It is approved by the different regulatory agencies. And the first thing which I want to show you here, that is these four formulations. One is the Vivitrol, the first one, and the another two formulation and third formulation. The third formulation, 65% of the population having the particle size of 60 micron, whereas the first, second, and third, Vivitrol and the two formulations, the 50% of the population having the micron size of 100 micron microspheres. So here I want to show you that the smaller the particle, the faster is the release. So here you can see in the formulation number third having the faster release than the Vivitrol and the first two formulations. And here it is the in vivo release studies. So this is actually matching, that this B and C is actually matching. So it is a very good correlation between the in vitro and in vivo profile. And the fourth one is the overlapping of the profiling, and how much is the shift of the formulation, time shift. It is shown over there. So basically, these type of formulations are basically for the microspheres and extended release injectable, long-acting injectables. Basically, USP4 apparatus is used, that is a flow-through cell. I will show you the video, very interesting video by Arvika, how this particular flow-through cell apparatus works for the microspheres and for the long injectables. It is very interesting video. It is a flow-through cell system, and there is a media which is inside the bottles. That is a dissolution media, and we have flow-through cell samples. Why we are using the flow-through cell? Because in ours it is mimicking their blood circulation. If you are injecting the microspheres inside the blood, so it is circulating inside the body. So similarly, here the media is going through the pump. Pump is taking the media, and then it is passing that media inside the flow-through cell. The flow-through cell's temperature is maintained through the heater. These are the various units of the flow-through cell. The media goes, there is a formulation inside, that microsphere formulations. It dissolves the drug from that, and then it goes back into the media again. So that the same media goes into the same bottle, and after a certain duration of time, like your dissolution times, like 1 hour, 4 hour, 8 hours, and do the sampling. That is the sampling points, and we monitor it, how much drug is there into this particular media, how much drug is dissolved. So it is completely mimicking our body system. So this is another, that is a sample collector, and then from the sample collector it will be injected into the HPLC. So it is all completely automation system. So this type of known as closed loop type of systems. There is open loop type of systems also, where it is not going back into the media. Whatever it is going through the flow through sample, we are collecting into the tubes and monitor it. Now, this is the same example of Vivitrol, which is through flow-through cell. So what are the regulatory guidance for the long-acting injectables? That is the current bioequivalence guidance in regard to long is that the generic formulations are required to be qualitatively and quantitatively be same as the reference listed product. This is very important guidance. If anybody is doing the generic development of long-acting injectable, it should be qualitatively and quantitatively be similar. Qualitatively means having the same excipients and quantity also. Then only it will be able to register a particular product. Now coming to the nanoparticles, that is the last part of my presentation. What are the regulatory guidelines which is going on with the nanoparticles? Still there are only 1 or 2 products which are launched across the world with the nanoparticles, but the lipid-based system is having the upper hand inside that, and that is basically liposomes. Various liposome formulations are there into the market and already commercialized. So if you see, it is categorized into the 3 types of system: polymeric, lipid-based, and inorganic type of systems. In the polymeric-based, it is gendrimers are there, polymeric micelles, polymerosomes, nanospheres are there. In the lipid-based system, liposomes, then emulsions, nanostructured lipid carriers, solid lipid nanoparticles are there. Then in inorganic, we have silica nanoparticles, carbon nanotubes, and metallic nanoparticles are there. So the formulation, these are the categorization across the worldwide for the by the regulatory agencies. And nano technology means the particle should be in the range of 1 nanometer to 100 nanometer. That is the common concept by the regulatory agencies. So what is the nanomaterials application? If we are doing this, it is generally if you are an organic chemist, or if you are the making some APIs or organic chemist, then you can remove this or expensive solvents with the catalyst. That is a green technology. So you can use some nanomaterials as a catalyst to replace the expensive solvents. You can reduce the contaminants such as lead, that is the heavy metals, through this nanomaterials to enhance the bioavailability for site-specific delivery, for controlled delivery, drug delivery, to decrease the dosage, means to decrease the drug requirement, API concentration per dosage, and decrease the toxicity of the drug product. So these are the basic objectives of using the nanomaterial nanotechnology products. And coming to the liposomes, which is one of the which I have selected as from the nanotechnology because it is commercialized and guidelines are well structured for the liposomes. So if you are making liposomes, there are very clear, lucid guidelines given by the regulatory agencies. Everybody knows what is liposomes. It is a bilayered structure made up of phospholipid bilayers, having the amphiphilic molecules, having the polar heads and non-polar tails, and which are organized into the one single layer, bilayer, or either the multi-layers. And the polar heads, towards the polar head, there will be some water aqueous space, and in the lower polar tails, there will be some your hydrophobic drugs will be encapsulated inside that non-polar tails. And then you can modify the surface of that particular liposomes by using some peptides or by using some materials which decreases the immunogenicity of the particular liposome, right? So this is the very important thing. And release from the liposome is basically depend upon the circulation half-life, which you can modify using the polyethylene glycol or cholesterol or other potential additives in the liposomes. And very important thing is liposomes are not filed with, as per the NDA, it will be through the biological license application. I will show you what is the difference between the two. What are the CQAs for liposomes? What type of test you need to perform when you are developed this particular formulations, and what your finished formulation should, what test you are going to perform. One is the morphology of the liposomes. That is how much lamellar determination, lamellarity determination should be there. For this, these are the regulatory guidelines. These are the requirements of the US FDA. What type of test parameters you have to do it. Surface characteristics of the liposomes. What are the net charge, that is zeta potential of the liposomes. Drug product viscosity should be given. It is very important. Drug product viscosity. If viscosity is very higher, then this lamellar structure, the liposomes, will collide with each other and become the bigger liposome. So this is very important thing. Drug encapsulation efficiency. What do you mean by encapsulation? That is entrapped drug versus the total drug in the drug product. If your entrapped drug into the liposomes is only 40% and of the total amount of the drug which is there in the formulation, that means your encapsulation efficiency is 40%. Then you have one more parameter, that is drug loading, that is drug to lipid ratio. What is how much drug is there into the lipid ratio or phospholipid ratio? Then we have the liposome phase transition temperature. How much is there? What is it? At what temperature these liposomes converted into the plastic phase, like from the solid phase, salt to gel temperature. Then particle size is very important. The mean particle size and the PSD, particle size distribution, both tests is very important. Then in vitro release of the drug substance from the liposomal preparation. That is in vitro release. I have shown you liposome, that is flow through chamber, flow through cell, that is USB4 apparatus. I have shown you the video. Leakage rate of drug from the liposomes throughout the shelf life. How much drug is permeated from the lipid bilayer structure from the structure to into the outside the liposome. Osmolality is very important parameter. If it is there, residual solvents. How much is the solvent which is left inside the drug product? In vitro drug release profile. Integrity changes. These are the very important drug release, drug encapsulation efficiency during the storage of the period. And in response to the salt concentration, pH and temperature. If slightly temperature there will be variation, how much is the drug encapsulation varies, how much is the drug release rate varies. So these are the very, by the external changes, by the external factor changes, like by the small concentration of this change in the salt concentration, how much is your formulation is varying. The integrity of the liposome is varying. If you have structure like multilamellar or 2 layers, and now after adding a slight pinch of salt, the salt concentration is varying, it becomes multilamellar, or the loading efficiency decreases. So these all parameters which we need to measure in respect to salt concentration, pH, temperature, or addition of some other excipients. And one and more very important thing, it should be sterile and free from bacterial endotoxins if it is for injectable formulations. These are the specifications for lipid components. We have to understand that the lipids which are there, it has to be structurally, we have to find out the purity of those lipids. The lipids, because liposomes is basically the raw material is phospholipids, or it can be synthetic, or it can be from the nature. Okay. So this we have to understand what are the, if it is how much is the trans fatty acid impurity there, free fatty acid impurities are there, peroxide impurities are there, lysophospholipid impurities are there. These impurities we have to identify into our lipid system. So this analytical procedure should be there, well-established analytical procedure should be there, so that we can determine how much impurities are there in our lipid system and how much during the stability is increasing. Structure alerts is there, that it should be for synthetic lipids and semi-synthetic lipids, and we have to do the testing to so that we can identify how much impurities are generated. Stability studies of liposomes, which is very important here to understand that we should address the microbiological, physical, and chemical drug product, including the integrity of liposomes in the drug product. We have to determine the size distribution of the product. Liposomes are susceptible to fusion. Therefore, we have to, during the stability, we have to see whether some aggregation or should not be there. And if it is there, how much percentage it is there, which is it should be under the acceptable range. And then we have to find out the chemical stability of the lipid components, which I have just told you. If you see, the unsaturated fatty acids are subjected to oxidative degradation, whereas the saturated and unsaturated lipids are subjected to hydrolysis to lysolipids and free fatty acids. So we have to find out the impurities of the fatty acids. This is very important parameter for filing of the liposomes. As I have filed 2 formulations for the liposomes which are under filing. So these all parameters are very important. SUPEC, there is no SUPEC guidelines for this liposomes, that is scale up post approval changes. So if you are doing some changes in the manufacturing process, or you are changing some concentration of excipient, so you have to do those studies in the laboratories and produce the data to the regulatory agencies to get the approval. So there is no as such SOPEC guidelines for the liposomes. This is about the biologics, which I have told you. The IND phase is very same, but in those we are in the phase 1 we are doing the immunogenicity studies, that is the safety studies, and then in the phase 3 we are doing the efficacy studies. Where in the new drug application we are doing the efficacy studies starting from the phase 2. So this phase 1 and phase 2 is on the human volunteers, the normal human volunteers, healthy human volunteers, whereas the phase 3 it is on the patients. So here is the difference. It is through the biological license applications, this liposomes. So I am very thankful to the local organizing committee, Shri Parveen Gargji, chairman of the college, Janesh Gargji, secretary of the college, principal and the director of the college, Dr. G.D. Gupta, R.K. Naran sir, the vice principal of the college, and organizing secretaries, Dr. Pooja Chawla and Dr. Vinit Rai, and all of the ICT teachers and family, technical and non-technical staff also, for organizing such a brilliant or good ISFCON 2021. At last, special thanks to the ISF family. Thank you very much. I think I hopefully make you to understand the regulatory requirements of the 3 dosage forms in this span of 1 hour. Thank you very much for inviting me on this particular podium.
Thank you very much, Dr. Gaurav sir, for such an exhaustive talk, which must be beneficial to all the listeners, delegates, and all the teachers. So to sum up what sir has discussed, the main thing which sir emphasized and motivated us that 40% of medicine that is supplied to US is from India. So that is a great achievement by us, as well as Dr. Gupta's. We are also proud of sir that they are going to manufacture Sputnik vaccine, that is a sort of Russian vaccine for COVID-19 in their Shilpa Medicare Limited. So congratulations to you, Gaurav sir, for this coming achievement. And sir very exhaustively told us about the evaluation parameter, synthetic parameter, regulatory affair for transdermal patches, liposomes, injectables, implants, which is the, I think, ask of the day. And like transdermal patch is very simple to apply, and they give benefit us for 3 to 4 days. So that is also very key point given by the sir. And sir also told that in all regulatory aspect, we should follow the ICH guidelines. Sir told us about the IND, NDA, NDA, BLA, and exhaustively told us about the difference between NDA and ANDA. And from our M form or B form, we study that drug discovery period take 14 to 15 years for the development of new compounds, in which the important phase is development phase, in which we use preclinical trials as well as clinical phase. So sir exhaustively told us the importance of preclinical as well as clinical phases, in which what drug regulatory affair we should follow. And he also discussed about the CDT, that it has a 5 modules, which may be very beneficial to all the listeners. And in transdermal patches, the main thing is that we can use animal skin as well as artificial skin to study the analysis or the drug release studies. So at the presentation and slides which was provided by sir, and especially videos, so the videos are very informative, slides were very informative, and I congratulate Gaurav sir for giving such a nice presentation. And I also congratulate all the ICF team, especially the mentor of ICF team, Professor G.D. Gupta sir, who has organized this conference in a very brilliant way. He is also our mentor and pathfinder. So thank you, Gupta sir. So I think what Pooja madam, will you please tell us?
Thank you, sir. Thank you so much. First of all, I welcome all the dignitaries who have joined us here. And Gaurav sir, your presentation was just awesome. The way you presented, the slides were, it really went into my brain completely. Though I am from pharmaceutical.
डॉक्टर पूजा
Yes, sir.
मैडम, हमारे
Yes, sir. Yes,
सेकंड चेयर पर्सन भी हैं डॉक्टर विनय चावला जी, वो भी कुछ प्रकाश डालना चाहें।
जी जी सर, बिल्कुल।
डॉक्टर
Sir,
चावला जी। हाँ, yes.
Good morning to all.
Good morning, sir.
Good morning, sir. It was indeed a very informative talk. Although I joined late, but whatever I could comprehend, that it was a very thorough and very illustrative presentation, and whatever slides that I could see, I could understand the illustrative aspects of each and every slide, and the presentation was really made very well. Congratulations, sir. It was a very good presentation. Thank you.
Thank you, sir.
So thank you very much, Dr. Gaurav Goel ji, and thanks to Dr. Sushil Kasab ji. And after long back, we are just on the, you can say, online mode as a video. Number of time we can discuss on the phone, but after long time we are talking, and this interactive session, and you have remembered the pharmacy family just like a part of your life. So we all are highly thankful to you, because if any person work to any place and after the move with the any region for the growth or development, so many things it may be possible. But if you can contribute your institute for the long time, it is always rememberable. So this is the very, very good thing. And thanks to all the organizing committee, and the technical sessions are going on very smoothly and timely. So thanks to all. Thanks Vinay Chawla ji, is the always available, because he is the person of the pharmaceutics, and he is also doing the good job in their universities. Dr. Amit Agrawal ji is also on screen, so I also welcome.
Good morning, Dr. Sir.
Good morning, sir.
Sir, am I audible to you?
Yes. And
Yeah. Sir, should we proceed for the next session, sir? I think, am I audible?
yes, you are audible. And am I audible? I'm
Okay.
Dr. Amit from PGI.
Yes, sir, you are audible. So moving towards our next session, we have Dr. Amit Agarwal, Assistant Professor at Department of Telemedicine, PGI MER. The topic of his talk is Telemedicine in Health Sector and its Future Perspective. And to chair his session, we would like to invite Dr. Vinay Chawla, who is Principal, UIPSR, Baba Farid University of Health Sciences, Faridkot, to please introduce Dr. Amit Agarwal. Over to you, Chawla, sir.
Thank you, Dr. Pooja. It's a pleasure to invite on stage Dr. Amit Agarwal, who is Assistant Professor in the Department of Telemedicine at the famous PGI Institute of Medical Education and Research, Chandigarh. Dr. Amit did his Masters in Biotechnology from University of Rajasthan, Jaipur, and doctorate in the role of T regulatory cells in asthma, especially in children, from PGI itself. He has also worked as scientist C in ICMR Advanced Center for Evidence-Based Child Health, and is also involved in many systematic reviews related to the emerging fields of medicine. He is actively involved in M Health project, which is funded by Bhabha Atomic Research Center, Mumbai, aimed at validating the tele-ECG device developed by BRNS, Mumbai. Dr. Amit Agarwal has to his credit more than 70 research publications in peer-reviewed medical journals and book chapters. He is a life member of different international agencies. He is registered stakeholder at National Institute for Health and Care Excellence, the United Kingdom, for asthma quality standards and cystic fibrosis quality standards. Dr. Amit has been awarded many international and national recognitions. He is the knowledge partner for EUPHC's 108 ambulance services and 102 services assessment with the Department of Health and Family Welfare, Andhra Pradesh. He is also involved in different policy making decisions with Ministry of Health and Family Welfare, Government of India, especially in developing the National Digital Health Mission Blueprint. So we are glad that such a great personality is among us. Dr. Agarwal, the stage is all yours, please.
Thank you, sir, for the nice introduction. And thank you for all organizing Secretary Dr. Gupta, sir, and Pooja, madam, for inviting me. I made a presentation I made related to the telemedicine role in the health sector. But I also want to touch some aspects related to the telemedicine role in pharmacy. So I am going to present my slide. So can you see my slides?
Yes, sir.
Okay. So the talk is on overall approach of telemedicine in health sector and its future perspective. So first of all, from my institute, that PGI-MER Chandigarh is a national importance and pioneer in providing both basic medical care as well as highly specialized quality services, mainly to the regions situated in north part of the India, Chandigarh, Haryana, Himachal, Jammu and Kashmir, and Leh Ladakh, some parts of Uttar Pradesh, Uttarakhand and Rajasthan. So these are the main headlines of my topic. Is like, what is the definition of telemedicine? What is the history of the telemedicine? The types of related to the telemedicine? What is the impact of COVID-19 and its on telemedicine? What we are doing in telemedicine PGI-MER, the future perspective, and telepharmacy. So first is, what is telemedicine? As you all know, that the term which telemedicine means we have to provide the healthcare delivery to the patient who is presented at a distant mode or in a rural areas or any area where the health services is not being approachable. So it is the term given to the remote healthcare delivery and consultations given to the patient at a distance. And this service is more accessible, cost effective, and it increases the patient engagement. It allows the healthcare professional to evaluate, diagnose, and treat the patient at a distance using telecommunication services. So telemedicine is all about the providing medical facility or healthcare delivery to the patient who are present at a distance. So what is the history in the telemedicine? So telemedicine is not a new practice. Like we thought that telemedicine is a term because of the COVID. We thought that this is a main era of the medicine, and very less people know about the telemedicine. But it is not a new. The concept of telemedicine is dated back to the 19th century. Okay, and what began as a few hospital wanting to reach patient in remote location, and it's become an integrated system across the care continent. So the history of telemedicine will unveil how we got to where we are today. So the history is like when the discovery of telephone by Graham Bell in 1876, the first effect, like because telephone is a mode where you can contact to anyone. So it's related to telemedicine. We can say that in 1876, the start of telemedicine by Graham Bell, discovery of the telephone, and launches the beginning of our telecommunication field. In 1924, Dr. Hugo Gernsback, he invented the teledactyl. Teledactyl means robotic.
Excuse me, sir.
Yes.
Sir, slides are not moving.
Not moving.
Yes, sir.
Oh.
Not changing, I mean.
You can't see the pictures of Alexander Graham Bell and.
No, sir. Only the first slide is visible.
Okay. So 1 second, I will just stop sharing and again I will try to.
Okay, sir.
Now it is moving?
Yes, sir. Now we can see.
Okay, please tell me that it is moving or not. I just want to switch 1 or 2 slides. Like this is the previous slide. Now it is moving or not? That telemedicine not moving.
It is still done on the Alexander Graham Bell and that slide only.
Okay. 1 second, I just called my technical person for that.
Okay, sir.
Slide is moving or not?
Yes, sir. Now it is moving.
Okay.
Next, please, just a minute.
Yeah.
Just to check out.
Now, okay, sir. Right.
Hanji, yes, sir. Right.
Okay, so I am on the slide of this that Graham Bell, the discovery of the telephone. It is visible now. It is moving.
Moving, sir.
Okay, so in 1924, Dr. Hugo Gernsback, he discovered teledactyl. Teledactyl means robotic fingers. And in 1950, medical personnel start doing experiment with closed circuit television. CCTV has been discovered in 1950. In 1960, NASA takes on telemedicine and is researched ways to provide healthcare to astronauts and improving telecommunication technology. In 1970s, the golden age of the telemedicine research and expansion, the federal government in US funded a range of telemedicine programs to improve healthcare access in the rural areas. And 1989 to 1999, the first time patient was successfully defibrillated by the telephone. So this is the past. Like now in the 20th century, video chat program like Skype and other apps take off. In 2000, telemedicine center in PGI also was established. Government of India launches National Digital Health Mission and Digital India Initiative in last year by the Prime Minister of India during the flag hosting ceremony on 15th August. Today, telemedicine is the basic basic service which is provided by the telephone or mobile phones and computers. Government of India also want to reach each and every patient which is situated in the rural and urban areas. So they established Ayushman Bharat Health and Wellness Centers for this cause. Home-used medical devices make it possible for the caregiver to monitor everything from vitals to glucose levels. And physicians can gather essential medical information, make a diagnosis without patient stepping foot in a doctor's office. So how many types of the telemedicine? Telemedicine are like 4 types. First is live video conferencing. Live video conferencing means two-way audio-visual link between the patient and healthcare provider. It is often used for the common illness. It is two-way, like provider to provider or patient to provider. Okay, and the second is store and forward. Store and forward means the person or patient come to the hospital or primary healthcare center. The physician see that patient physically and send the reports and all the diagnosis to the specialist center. And it is store and forward because there is no live communication has been there. So it is a store and forward basis. Third is remote monitoring. Remote monitoring means patient has to record their ECG, their spirometry, their other medical issues or data in an electronic tool and forward this to the provider, to the other location. So this is the remote monitoring. Fourth is real-time interactive service. Real-time interactive service is required in the emergency patient where immediate advice to the patient needed. So COVID-19 and telemedicine. So you can see that telemedicine is very much helpful in the COVID-19 scenario. In PGI, in the shutdown of the OPDs and other hospitals, tertiary care hospital are shut down due to the COVID. Telemedicine is a tool where the prescription, the diagnosis has been made to the patient and problem can be solved by this telemedicine platform. And we all are using the telemedicine or the tele-education by conducting the webinars, the seminars, the conferences are online. So telemedicine is very helpful in this COVID era. And telemedicine is very much positive contribution in healthcare during this pandemic in varieties of ways. During this global pandemic, telehealth is emerging an effective and sustainable solution for precaution, prevention, and treatment to stem the spread of COVID-19. So one quote is there that by deploying telehealth solution and programs, people who are suffering from other medical ailments during this time can receive care from home without entering medical facilities and minimizing their risk for contracting the virus. Means the patients who don't have COVID, but they have other medical problems, they can contact the medical doctors and medical professionals through telemedicine without entering the medical facilities. So telemedicine also can help first responder in the field communicate with doctors, helping to ensure those who need hospital care get it quickly and efficiently, and at the same time divert those who don't need any hospital care to other facilities and keep them their home safe their home. Like if any patient need any medical attention, he can contact to the telemedicine facility, and doctor can decide the medical attention is required at the emergency basis, or the patient don't need to visit the hospital. So by deploying advanced telehealth solution, physician can expand their reach even if quarantined, with the ability to remotely examine and diagnose more patients in a shorter amount of time, and minimizing the number of patients entering hospital and medical facilities. So these all are from the telemedicine perspective. So what are the benefits to the patient? The most benefit to the patient is related to the underserved rural, semi-urban and remote areas where healthcare services are very low or not up to mark. So the patient are very much benefited to that areas. Access to expertise of medical specialists to a larger population without physical referral. Reduce visit to specialty hospital for long-term follow-up care for aged and terminally ill patient. Means the patient who has aged 70 years, he can't walk to the healthcare facility. He just want to just repeat his medication on the card. So he does not want to visit the hospital, just do the consultation by the telemedicine, and the follow-up can be continued. So what is the benefit to the physician? It is improved diagnosis and better treatment management, access to computerized comprehensive data of the patient offline or online or in the real time, quickly and timely follow up the patient discharge after palliative care, and continuing education on training through video conferencing periodically. So hospital and insurance benefit. Hospital, the most important benefit to the tertiary care hospital like PGI is that unnecessarily visit of the patient or the burden of the patient is decreased, significantly decreased due to facility of the telemedicine. Earlier discharge of the patient leading the shorter length of the stay, increase in the scope of services without creating physical infrastructure in remote hospitals. So the most important factors in the telemedicine, like no transportation cost involved, no need to take time off the work. You can talk to your professional staying at home. Eliminate child or elder care issues. They don't want to visit or they need not to visit to the hospitals. On-demand options are available. You can consult to your where you want to like to the professional or the doctor you want to chat with him or to consult with him. Access to the specialties, like anyone in the Haryana, Punjab and Himachal is situated in these areas, he or she can go to the ISFCON Health and Wellness Center and reach to the PGI MER by these facilities and they don't need not to visit PGI for this consultation. Less chance of catching new illnesses, less time in waiting room and better health. So Government of India provide a very grateful platform eSanjeevani for this kind of consultations, and this eSanjeevani platform is developed by CDEC Mohali. So it is very much privileged that it is developed in the Tri-City, near to Chandigarh. So two type of eSanjeevani platform has been made: Health and Wellness Center platform and telemedicine OPD program by eSanjeevani. And if you listen to the FM or radio, you repeatedly listen that you have to register to the eSanjeevani program, and there is OTP in your mobile, and then you have to consult with the doctor. So this facility is because of Government of India initiative, and it is very much helpful in this pandemic scenario. So two type of, as I already said, that two type of telemedicine platform is there, health and wellness centers, that I already told you that you can visit any healthcare wellness center and it is connected to the PGI. You can contact to the PGI specialist doctor if there is any emergency or if there is any specialty consultation required. And you have to, if you just want to do OPD consultation, you can just reach out by installing the app on your mobile phone, and it is all there. For this management, Board of Governors of that governing body of the Government of India released the telemedicine practice guidelines so that RMPs can do the consultation easily. So what is the role of Department of Telemedicine, PGI Chandigarh in COVID era? We have in just 3 months, April 2020, May and June, where when COVID is on the start phase, or people are very much fear in that to visit the hospital, over 50,000 teleconsultation has been provided by our department. CPC and CMEs webinars in very regular phase. Institute MD, MS, DM, PhD exams are conducting from this department, and classes of various department has been conducting. So these are some screenshots of different departments organizing their education and learning programs from PGI, from telemedicine department. So why we need to expanding the telemedicine? There are two main areas of adoption for telemedicine at this time. First, that hospitals are expanding their telehealth services, and second, they have to find the ways to train the staff. So telemedicine is very much promising findings to train the staff remotely. So what is the pros and cons? Like if we can see that limit exposure of the patient and healthcare for the SARS-CoV-2, it can preserve the PB, efficiency tries patient in need to urgent in-person evaluation, expedite the time from referral to evaluation and listening. Cons are there, that difficult to build rapport with the patient, limited physical examination, because you are not seeing the patient physically, you just see the patient on video conferencing, difficult to observe interaction among family members and other caregivers, not suitable for all patients, like very ill or difficult with the technology. It is you can't see the patient with the technology, uncertain feasibility and sustainability following COVID-19 public health emergency. So what services can be provided by telemedicine? First, primary care and specialist referral services. Primary care and specialist referral services means that health professional providing a teleconsultation with the patient, where the patient already has been seen by a physician who is present at the primary healthcare center. So if the physician who is presenting at the PHC, if he see any problem with the patient and he needs some consultation from the super specialist, he may involve to use the live interactive video session, or he can send the images of the patient and its report for store and forward basis consultation. Second, remote patient monitoring. As I already said, if patient have the facility to record his physical examination like heart ECG, blood sugar level, he can send the reports to the physician who is present at the tertiary care hospital, and the doctor can also see the reports and advise remotely. Third is consumer medical and health information. If anyone need any health information related to any kind of hospital, the information made available for with the help of telemedicine. Fourth, if the patient is not able to visit the healthcare facilities, or they are not in a position where the healthcare facility is very far from their home, he can easily approach to the nearby healthcare center and do the teleconferencing with the tertiary care hospital. It improves the medical education. As I already say that the seminars, webinars, the healthcare professionals, then they can train their medical staff, their medical education by conducting the training sessions from the telemedicine. As you saw that in the picture, the CRP method and how to give the oxygen to a patient, that the training is going on. So medical education is been there. So what are the advantages of the telemedicine in perspective to the cost bearer, to the physician, to the patient? Okay, so for the cost bearer, the higher quality of life, like disease management easy hai uske liye, there is reduced patient periods of hospitalization, transparency, the cost and services quality assurance is there, lower medication cost. So cost is very much decreased from in perspective of patient or hospital. Patient improved compliance, changing in the lifestyle and acceptance of the disease. Means he has to understood that when he has to visit the hospital or when he does not want to or does not need to visit the hospital. So in the physician, the security, quality of life is increased, therapy guidelines and control, budget relief and optimized time management. He can see more and more patient through the telemedicine by sitting at his clinic or by sitting in his room, direct guidance and training of the patient and optimization of the therapy. So telemedicine has been growing rapidly because of the 4 fundamental benefits. It is improved access, cost efficiencies, improved quality, and patient demand. So improved access means for over 40 years, telemedicine has been used to bring healthcare service to patient in distance location. Not only does telemedicine improve access to patient, but it also allow physician and health facility to expand their reach beyond their own offices. So telemedicine is like improve the accessibility. Second is cost efficiencies, like it reduce the cost mainly in the perspective of patient. Their travel time, their travel cost, they don't need to leave their offices. They can go to the offices and earn because in the India, the people are related to their daily wages. They don't need to lose their daily wages because it is very hard for them. Like so, the cost efficiencies is better in that perspective. Third is improve quality. Because of the care, because all the diagnostic, all the reports come to the hospital in a proper manner, it is the professional, the physician can see the reports by relaxing mode, not in a hurry mode. So quality of life, quality of the medications, of the consultation is very much increased through telemedicine. Patients to demand service. Now we have conducting a survey related to the patient satisfaction during the telemedicine, and we have found that 70 to 90% of the patients is very much agree that this kind of service may be continue after the pandemic ends. So patient also demand to they want to do telemedicine services because it reduces the travel time and stress of the patient. So rise of the telemedicine, like rinsing use of telemedicine takes different forms. Traditionally, telemedicine has played a biggest role in the rural areas. First, like I want to say that now it is used in urban areas also, not not for the rural or distant areas. The urban areas where the aged people, the people who are like in the they don't have their caregiver with them, so they have no need to travel or no need to visit the hospital, and the patient can be connecting by the best expert. They can choose the expert because there are different digital platform available on your phone, so you can just see who is expert on that field, and you can just click on that doctor, pay their fees, and you can do the teleconsultation services. But it is for the private purpose. Telemedicine facilities for the PGI, it's free. It's not any cost involved. So tele-education is very much important, and it is related to, and we are also in the Department of Telemedicine, we are involving many artificial intelligence and machine learning technology to provide tele-education in the best way and learning possible for any potential recipient. So, but there are some legal and ethical issues related to telemedicine. To overcome for this telemedicine problem, Government of India has come with the telemedicine practice guidelines, and they can see that patient can call to the doctor directly. So when the patient call to the doctor directly, the consent is implied, means patient wants to consultation, and there is second type of consultation like doctor to doctor. No consent is involved because doctor already see physically the patient. But there is a problem in cross-border teleconsultation. Like if any patient situated in the Nepal hospital, he want to consult with PGI, so there is some ethical and legal issue also involved in during teleconsultation or telemedicine services. So I just want to say that you can use your smartphone to see the retina, to see the ECG, to see the spirometry. Everything is your phone, and it is also a part of telemedicine. So this is overall effect of, like I just told you, in healthcare sector. I know that this talk is very much of the clinical perspective, and the presentation or the target audience is like on the pharmacy field. So some few slides related to telepharmacy I just want to address in this talk. So telepharmacy. Telepharmacy is the delivery of pharmaceutical care via telecommunication to the patient in location where they may not have direct contact with pharmacist. Actually, in India, the pharmacist or pharma people, they are just stick with the medical stores. They don't have very much positions in the hospitals. But in foreign countries, pharmacist plays a very important role. Without any prescription which has not been passed by the pharmacist, you can't buy any drug from the medical store. So telepharmacy is like it is very important in that way. It includes drug therapy monitoring and patient counseling with the use of teleconferencing. So implementation of telepharmacy is very much related to the region and jurisdiction, and factors including geography, laws and regulation, and economic influence and its implementation. So telepharmacy is very much new in India, but in the countries in the Western countries, it is very much in use. So I just want to tell about the different countries, how the telepharmacy is going there. So in Australia, the Royal Flying Doctor Service, the telepharmacy is name given like Royal Flying Doctor, and this service is since 1942. It has been started in 1942, and they have made packets like medical, and their name is like medical chest. So medical chest, it contains the medication, the equipment which are placed in the remote center, and they can be administered to the patient during a telehealth consultation. And 3,500 chests were distributed around Australia as of 2006. So Queensland is a state in Australia, recorded 21,470 telehealth consultation in 1 year, and the medication administered mostly antibiotic, analgesic, and gastrointestinal medication. And this system is improved to in the both emergency and routine medical care in remote parts of the Australia, and reduce the need for the patient to travel to seek medical care. So if I talk about the United States, the telepharmacy began in 2000 and the need of the delivering pharmacy services in the mainly the problem with the rural communities. So in 2001, North Dakota became the first US state to pass regulation allowing retail pharmacies to operate without requiring any pharmacist to be physically present. Or in 2002, telepharmacy project has been established. In Canada, the first telepharmacy was started by the hospital in Cranbrook, British Columbia, and remote dispensing machine for medication began operation in Ontario, Canada in 2007. What this machine do, that patient insert a prescription into this dispensing machine, the prescription is scanned and the patient connected via the telephone or video conferencing to the pharmacy store and get the prescription prescribed medicine there. So what are the advantages of the telemedicine? That in all the countries that I have mentioned, they have significantly decreased the number of adverse drug events reported in this country after implementation of telepharmacy. It's saving like several thousand deaths each year. And the primary advantage of telepharmacy is the easiest access to telehealth services in remote and rural areas. It also has the several economic benefits. Like it is very much needed in India because 60% of the patient in India have the difficulty for the affording the medication. Like doctor just write the medicines, but patient doesn't afford to buy the medicine. If their travel cost or their other expenses can be reduced, he or may be available for to buy that medication. So it saves the travel time, expenses, and it is a major barrier for rural, elderly, and disabled veterans. So just conclusion of my talk is that we must contribute to limit in-person patient care where it is appropriate to avoid a third wave of SARS-CoV-2 transmission. Even when in-person clinical visits and procedure are an option for most patients, many will still prefer telemedicine alternatives to in-person care because they all now they are fear of coming to the hospital. Patient will largely dictate when they are ready to return to the business as usual. Don't know that when the pandemic ends. So telemedicine continue to be an important part for providing the teleconsultation. And telepharmacy is quickly becoming an integral part of modern pharmacy practice. And it has a potential to provide quality pharmaceutical services such as medication management, dispensing, patient counseling, and drug information. So if we talk about like telepharmacy, and if we thought about the telepharmacy, and if there is some artificial intelligence, machine learning is involved in the telepharmacy, or we can make some application related to that, it is really a very good opportunity to all the pharmacy students to come in the telemedicine field. So for these words, thank you. And I also thank to all the organizing secretaries and the chairperson sir and director sir for inviting me for this talk. I know that this talk is related to, and I repeatedly tell that this is related to the clinician, but is very much helpful in overall perspective at how you can reach out with the telemedicine.
Thank you, Dr. Saab.
Thank you so much, Dr. Agarwal. Now I invite Dr. D.N. Prasad to please conclude the talk of Dr. Amit Agarwal.
First of all, I would like to thank Dr. Amit Agarwal, who have given a very informative talk on this topic. And it is very helpful for practitioner as well as pharmacy people. Both have been very much coordinated by Dr. Amit Agarwal. And Dr. Saab has broadly explained about the telemedicine. He has explained about the history of the telemedicine, what is the definition of the telemedicine, what are the types of the telemedicine. Or in this case, we can say that no doubt, as we have gone through the entire talk of the Dr. Agarwal, We can also say that it is a digital modern therapy technique. First of all, I would like to share the talk of this Dr. Agrawal about the types of the telemedicine. He has explained about the 4 types of the telemedicine. That is the video conferencing, store and favor, remote monitoring, real-time interactive services. So the entire types covers all types of patients, even a normal patients who are suffering from a normal disease, or even the patient who is very much serious. As for example, real-time interactive services provide the emergency patients. Now, Dr. Agrawal has also explained about the benefit of the telemedicines, how the telemedicines gives the good health services to the, we can say, the larger populations who are living at distant positions. In fact, there is no wastage of time if we use the telemedicine therapy. There is an excellent interaction with the patients who requires a treatment. Dr. Agrawal has also explained about the policies of the Government of India. The Government of India has given some platform. Dr. Agrawal has explained about the e-Sanjeevani telemedicine platform. And he has also given the example about the Tri-Cities, where it is very much active. Nowadays, there is a COVID-19 pandemic, and the telemedicine's OPD has been opened. And this telemedicine is also very much effective in controlling the COVID-19 patients. Then Dr. Agrawal also make aware about why the telemedicines are required, what are the advantages of the telemedicines. And the main thing is, in fact, we are the pharmacy people. And he has also explained about the telemedicines, which is no doubt it is a very new concept in India. Dr. Agrawal has also made us aware about the role of the telemedicines in different foreign countries like UK, Australia, Canada, where medical chests are available. Or in case of medical chest, no doubt we can say that curtails the time of the treatment. And he had also explained about the advantages of the telemedicines. What are the different advantages of the telepharmacy? As for example, there is a adverse therapy reports is minimized. Low income people are also benefited. It is a time-saving, we can say, treatment. So Dr. Agrawal has covered almost all the topics, all the parameters regarding the telemedicines and telepharmacy. And I hope that the viewers take the advantages of this talk. So then I would like to thank Dr. Amit Agrawal again for giving a very valuable informations about the telemedicines and telepharmacy to all the viewers. Thank you, Dr. Sir.
Thank you, sir. Thank you, sir. Sir, there is a question from Dr. Saurabh.
Saurabh Kose. Yes, sir.
Yeah. He just want to ask something. So please.
Kose, sir.
Yeah, good morning, Dr. Agarwal. Indeed, it was a very good talk. As a pharmacy practitioner and academician, I have sir 2 questions. What will be the impact of telemedicine in rural population as far as India is concerned? Because as cross compared to the, as you told sir, Tri-City, and in the metro city or big cities, it is usually applicable. And way back 10 years back in New Delhi, Aam Aadmi Party Mohalla Clinic was also started with the same concept, medicine vending machine. So indeed, sir, it is very useful. We have seen in pandemic period. So just I have a concern over the rural and the tribal populations of India.
So I just want to answer your questions. It is very good question. Actually, in my presentation, I already talk about the health and wellness center. The concept of mohalla clinic, or concept of, as I am not the knowledge partner of EUPSCs in Andhra Pradesh. So I visited that kind of setup, and I have visited mohalla clinic also, and I have visited a health and wellness centers. So all the centers, they have the similarity of like telemedicine. What has been there that in the rural areas, no medical hospital, big hospital is there. So there is like panchayat hospital type, like primary healthcare center, secondary healthcare center, or community healthcare center. So government of India targeted approximately to develop 1,50,000 health and wellness center all over India. And we are connected with the Haryana, where 30,000 health and wellness centers has been developed, and we are connecting, we are providing, every day we are providing like 2,000 consultation in Haryana by PGI telemedicine center. So it is now it is developing, and definitely we will be developed in every rural or villages, and we definitely connected to the secondary and tertiary care hospital, so that each and every village can be benefited for this telemedicine facility.
Thank you so much, sir, for your answer. And definitely, this is one of the best session of the conference as far as clinical and pharmacy practice integration is seen. Thank you so much for your session, sir.
Thank you. And seriously, there are very much opportunities. If you implement the telemedicine with the pharmacy, very like much opportunities can be you can raise. So if you think about that, and there are a lot of posts, lot of advertisement is been advertised through our telemedicine department. Like now we have approximately 50 or 55 requirements, and we just posted in the PJ site. So persons are enjoying, yes, pharmacy persons can join that.
Thank you so much, sir. Thank you.
Thank you so much, Dr. Amit Agarwal. As Kose sir said, it was really a very, very informative talk, and specifically in the time of this pandemic, when we are anticipating and we are scared of the third wave of COVID. Thank you so much, sir, for sparing your time, your precious time from your busy schedule. Thanks a lot.
Thank you.
I would like to thank Dr. D.N. Prasad and Dr. Vinay Chawla, who were the chairpersons for this session. Thanks a lot, sir. Now, with your permission, I would like to move towards our third session for the day. Mr. Ranjit Bhashikar is the speaker for the day, and he is the CEO QBD International, QBD CGMP Consulting, United Nations Advisor, Geneva. And for the chairperson for this session is Dr. Sushma Talegaonkar, who is Associate Professor at Delhi Pharmaceutical Sciences and Research University. Welcome, ma'am, and welcome all the dignitaries once again. I would request Dr. Sushma Talegaonkar to please introduce our speaker, Mr. Ranjit Bhashikar. Over to you, ma'am.
Okay. Thank you, Dr. Pooja. It is my great privilege to share the dais with the Professor Bhashikar, who is a very renowned quality by design expert. I have read the biodata, and I came to know he is having a very rich experience of 51 years in a very diversified field of the pharmaceuticals, like manufacturing and US FDA and R&D, so many part of the pharmaceutical companies. And he is having a 51 years of experience. Currently, he is a CEO, Quality by Design International, CGMP Consulting, Biopharma, United Nations MPP Geneva Advisor, member of Editorial Board of Journal of Generic Medicine, England. He is a very recipient of so many awards. A few awards I will narrate. He is a recipient of the Quality Champion Gold, Topmost Quality Award by the Government of India, Quality Council of India. He is also the recipient of 100 Most Impactful Healthcare Leaders Global Award by the World Health Congress. He is also the recipient of Pharma Ratna Award by the largest NGO, Rabdi Meher. And he is also the expert on the quality by design, data integrity, CAPA, quality culture, and the various other current good manufacturing practices. He is the expert auditor of the formulation and API facilities and the clinical research organization. He also worked with the various multinational companies like the Pfizer Limited, Sun Pharma, Laka Labs, Cancer Research Institute, Mumbai, and he is retired as a global quality head, vice president in 2006. He also traveled globally in 70 countries and delivered 4 presentations in the conferences, chairman, and the keynote speaker. And he is also organized by all the conferences and the seminar is organized by the ISP FGA, Marcus Evans, IQPC, IPC, CBI USA, WHO, ISO, UNOPS, Government of India, CPHI, FGA of the Gujarat, Maharashtra, FGA Andhra Pradesh, FDA Karnataka. He also addressed, it is a very commendable, he addressed 42 webinars in the last 10 months in the pandemic situation in the various pharmaceutical subjects. His all articles, interviews published in the various international journals like the Pharmaceutical Manufacturing USA, Pharmaceutical Technology Europe, and Pharmaceutical Technology Asia digital version, to mention few. He was nominated as an API expert Geneva for the new ICH Expert Working Group Geneva on CTD QoS, served as a member of IPA, IGMA, ISP Gujarat Council of Biotechnology. He also worked as a faculty member of OPPI, IGMA, ISO workshops and was a member of GMP committee for API by DCGI Government of India. He is also the member of advisory committee, quality expert in SVK BNCP College, Mumbai. He is also the member of advisory committee, Pharmacy College, Talegaon and Nerul. He is also the mentor of Gitanjali University, Udaipur, student chapter, International Society of Pharma Economics, Outcome Research, SPOR. The biodata is so vast, although I have narrated each and everything. And now he is delivering a talk, a very vibrant and a very, say, the integral part of the pharmaceutical industry, that is the QBG opportunities and the challenges. And this quality by design, I would like to say, it is one of the most vibrant and the very required, we can say, the part in the pharmaceutical development, manufacturing, pharmaceutical analysis, and to maintain the quality of the product. So, and we all know it is a concept of the Juran, Joseph M. Juran was this quality concept he has introduced. And the main focus of this talk will be to how to maintain the quality and how to make our customer satisfied. Until unless we are not satisfying our customer, there is no use to make the product. So with these words, I will invite Professor Barshikar to deliver a talk on the QBG. Professor Barshikar, over to you. Namaste. Sir, you are mute.
Thank you. Thank you. Okay, thank you very much, Sushma ji, for such a lengthy introduction.
Sir, you deserve.
It's a brief, and usually I expect only 4 or 5 sentences out of that, but I appreciate
No, sir, it is highly motivating to all participants and all the scientists whosoever is attending. It's a great pleasure to invite and introduce you, sir.
Pleasure too. I'm thankful to all of you for inviting me to this wonderful ISF Moga College in Punjab University function as a keynote speaker today on quality by design. As such, this subject, Sushma ji, is very, very big. And the time of 45 minutes to accommodate this, I'm going to have a big challenge today. In addition to the implementation of TV challenges, need to maintain the time also is a big challenge. And let's see how it goes. In case there is any problem of audio video, I'm going to show you some videos also. If there is any issue, please speak out. Okay, send the messages on the chat box whenever you have any doubt. So I proceed now with the presentation. Are you seeing this?
Yes, sir. Yes, sir.
Now this is full screen.
Yes, sir.
Okay, great.
Yes, sir, very much.
Thank you so much. So this subject, quality by design, is important from the perspective of the product development. Product is robust to ensure that the product is having minimum defects, and to try to achieve with the help of quality by design the Six Sigma implementation over the period in phased manner. So what happens here? This is a disclaimer. So why this quality by design concept has been introduced by the regulatory agencies, especially a lot of initiative was taken by US FDA, because there were severe shortages of the products in US market. The consumers, patients were suffering, and they decided that let's find out the causes of shortages in the market. And they found out that there are severe recalls of the product in the market, and these recalls and the shortages have come out due to three major issues. One of them was quality issues of the product, lack of efficacy, and also the side effect. So keeping in mind these activities from the shortages perspective, they decided that we should address this from a different context. And then they further realized that many of these processes are not robust. There is no consistency in the processes, and there could be the various reasons for that. And these, some of the reasons from my perspective, is haste to file the product in US market. First to file has tremendous advantage, and you get 180 days exclusivity if it is para for filing. We don't spend enough time in development. We rush. We want to have shortcuts in product development. We don't invest much in research and development activities. And are these reasons for non-robust process? Every organization has to look from that perspective. Also, there are several issues from the perspective of the variations or the variabilities in material supplies from the supplier, issues during manufacturing, process manufacturing, and at the same time during the testing as well. There are so many variabilities. So if the input is garbage, output also going to be garbage. So these so many things have realized, and they thought that we must now focus on patient, focus on product. Let us design a process which is based on science and risk-based to ensure whole product life cycle. What the product life cycle means? It means not the shelf life. It means when you introduce the product into the market, and till the time it is in the market, I'm talking of product, not the batches. So it could be 10 years, it could be 20 years, it could be 50 years or 100. So whole So many years is the life cycle of a product. In other words, the product is in the market till the last day of the withdrawal, the product from the market, and you are discontinuing the product is a life cycle of a product. So during this period, it is essential to continuously improve the product in the interest of patient. And this is where we are talking of handling the variabilities, monitoring the variable process in a right perspective. And that is why the US FDA and subsequently ICH guideline Q8R2, ICH guideline Q8R2 has an extremely good definition on quality by design. And it says it's a systematic approach to develop a product. It says we begin with the predefined objectives, means what customers want, what patients want, and ensure that we enhance our product and process understanding and process control during manufacturing, which is based on sound science and quality risk management. So understanding of these, each of these heavily loaded words is important while implementing quality by design. So definition is, every word in this definition is highly loaded. There are several applications for this quality by design. It's API manufacturing, API development, API process development at different stages, in formulation development, and in analytical method development as well. So overall, these also can be further implemented in bioequivalence studies and clinical trial as well, in addition to what I mentioned just now. So this is a game changer for the industry in the interest of the patient, consumer at the end, and who should be always satisfied with the quality of the product, and they should not starve the product because of shortages in the market. So this concept has helped to reduce the shortages. So how do we move forward for that? From that perspective, as a roadmap, if you see this, we start with quality target profile, which is based on the expectations of the patient. Identify the critical quality attributes which are having impact on the patients. Similarly, identify critical material attributes, critical process parameters, and have a design space to have a right control strategy throughout the life cycle. So in short form, it is QTPP, CQA, CPP, design space DS, control strategy CS, and life cycle management LCM. Just you have to remember this short form throughout the life as you are in the pharma industry. One of the key supporting and nucleus of all this implementation is the risk management process. And this risk management process is, as I mentioned, it's a nucleus of QBD development. And rather, in every activity in the organization, also in your universities and colleges, the concept can be implemented to avoid the failures of the processes, of the procedures, of the activities. Even just like you are organizing this program now, webinar series. So what can go wrong in throughout the 3 days between 1st July and 3rd July? You are well prepared to handle it. What can happen suddenly if there is a power failure? What happens? So you have generator. So you have decided that I will quickly switch over to generator to ensure the continuous power supply. So same thing happens in the product development and during manufacturing and testing of the product. So there are three major areas to handle: is risk assessment, how to control the risk, to minimize the risk, and to review the risk in future by using different tools for handling the risk management. Some other time when I'll speak, if required, on how to handle this in different ways. But only today you keep in mind, what can go wrong? Where can I go wrong? Identify this in advance and mitigate it by having different controls on that to avoid failure. Next one, which is most important, though this is an option by the regulatory agencies, is the design space. It is nothing but the range in which your product can comply fully, and based on which you can have your own daily specifications for all critical parameters, critical processes, critical CQAs, quality attributes. So here I have shown you this normal operating range within the design space. And if you are following this, you are ensuring that my process will never fail. So I am predicting when my batch is within this normal operating range, I am going to pass the batch. In any case, everything will be fine. I have nothing to worry. And I can change this normal operating range to another range, provided it has more advantage than today's ranges. And if you want to go out of these ranges for sufficient and solid concrete justification, you can approach to the regulatory agencies. Without their permission, you cannot go out of design space. But within the design space, you don't need regulatory permission. You do not need. And once you have the design space and confirm the normal operating ranges, there are several models, there are several technologies by which you can control these processes and quality attributes. And one of them key is process analytical technology PAT. Process analytical technology. Remember, this has two major functions. One is handling the process, monitoring the process. And second is analytical monitoring. So both together is done at a time. And there are four alternatives to that. One is you can have the probe of the PAT machine to sense the reaction, non-touching to the product, and it gives the results and the procedure moves ahead. Then it's online, you draw the sample out, goes through the machine automatically, the results are printed, you move to the next step immediately without waiting for sampling and sending two samples to the quality control and testing afterwards, which takes few days or few hours. Here you can immediately switch over inline or online to the next step. So offline, your equipment is in manufacturing block. It goes, you are manually drawing the sample and sending it to the offline testing. In the quality control, in the manufacturing block, it is known as at line. Remember. So what we are doing by quality by design is transitioning to minimize the variations, to minimize the variability of input material, to minimize the manufacturing process variability and testing process once the finished product is ready. So it is helping us to transition from regulatory perspective compliance only to enhancing our product and process understanding and real-time quality assurance, real-time analysis, real-time data available to us. So in other words, from raw material to product, in other words, raw material to product. Once your product is ready, and throughout the life cycle, we have to give this information back to R&D, which feeds forward the data on CPPs and CQAs to ensure that product development, to ensure the product development is perfect throughout the life cycle and improve continuously. So when you develop the product like this, what is expected by regulatory agencies? The US FDA says, where I attended one of their programs, and I could pick up these slides from that presentation, is how we are handling critical quality attributes. How do we identify, are these right? Is the justification correct? And if we don't give this data correctly, they come back to you that justify these CQAs. We are not convinced. So it is essential for research and development to ensure the right information and right justification to be fed in the filings. Same thing happens while justifying this in a tabular form. We have to give the clarity to them that what kind of quality attributes we are addressing, QA, and what is the target of that with the justification. So this justification is completely with the risk-based assessment. Same thing further with the help of risk-based assessment, summary of the risk-based to rank the risk involved quantitatively or qualitatively. Here it is qualitatively, the risk is low, medium, and high. We have to address on priority the red blocks. For example, the product CQAs impact of the process and the excipient during manufacturing. So which are those excipients will affect which CQA of the product? We should identify that initially and then give them information about amended risk assessment, that how did you address it. Same thing if we are having active substance or API used in the product which are the different polymorphs. So we have to give the data to them whether it's polymorph A or B or C. The active one may be C, then it will not go to any other form throughout the shelf life of the product. Remember, this is the shelf life of the batch. So we have to give that justification to them. So then how do we handle this further? And that is where the detailing of this whole process is based on the different guidelines issued by ICH. Q8R2 is the latest version of pharmaceutical development. Q9 is quality risk management. Q10 is pharmaceutical quality system. Remember, Q8, Q9, Q10 are hand in hand. We have to read these guidelines together and to have a right interpretation. Q11 is for drug substance development and Q12 is life cycle management. Q13 is for continuous manufacturing and it is in the process of making the document, and Q14 is for analytical validation and QBD analytical process development. And I'm going to speak to you more on continuous manufacturing few slides later. So FDA thought that let us introduce this 21st century CGMP guideline risk-based approach, which will help the quality system with the help of PAT to monitor whole process, to monitor the critical process parameter and the critical quality attributes perspective. So it is very important when we want to go to quality by design, a mentality of the organization from top to bottom has to change. Though there has to be fundamental shift in the thinking of the organization, cultural change in the organization to implement quality by design, to ensure that we will mitigate the risk either by zeroing the risk or minimizing the risk to the maximum level by different strategies. And one of the area there is culture of quality has to be introduced in the organization through all the departments. Some other time I'll discuss with culture of quality to you and what is that operational excellence required to achieve this whole concept of the fundamental shift in the thinking. Operational excellence will also help us in Six Sigma achievement of the process. To achieve these desired results, of course, we need to invest into the development process, and right quality system is required from continuous improvement perspective. Of improvements are quality, the speed, and the avoiding the wastages to save the cost. And as we go along, you will see how we are achieving this whole process of saving the cost, of saving the wastages during manufacturing. Even 1% if we save of the batch manufacturing, it amounts to thousands of fresh package into the market to save the cost of each of the boxes. So to improve or to make quality better, to speed up the operations, to put the batches faster into the market, to launch the product faster into the market, and to have a lower cost of manufacturing, it is essential to move forward from improvement perspective. So it is a paradigm shift from blind compliance to science and risk-based compliance during manufacturing, during development, during manufacturing, and during testing of the product and release of the product to the market. So what happens then? Of course, for anything new, initial cost is always high. When you want to buy any machine, there is always a cost on day one. But as you use the machine to the maximum capacity every day, the return on that investment comes very fast, and the time comes, you achieve that desired state where the cost of that machine is fully recovered, and what you get afterwards is a bonus. So the tremendous cost saving as you follow quality by design over the period, from a current state of traditional manufacturing to the desired state of the quality by design compliance, where we achieve continuous improvement. Earlier in traditional manufacturing, even in many organizations are still on quality, rather traditional manufacturing today also. And they have a fixed formula, fixed process, and they are continuing to do that without ensuring the failures into the process. So this quality by design is helping us tremendously to have flexible processes and continuous improvement in manufacturing by addressing the CPPs and CQAs. So biggest advantage we get is the patient safety by way of right efficacy, availability of the product in the market, safe for use, and timely availability. Scientific understanding of the process to the company, scientists. Now you are giving a scientific justification that my design space is perfect and I'm using it to release the batches, so that you are predicting the quality of the batch when you use design space. To have a robust product, and robust process development, and thus it is giving us advantage of complete transparent operations in development of the product, in manufacturing and testing of the product before it reaches to the patients. So from the opportunities perspective, more you can see there is a process efficiency to help to achieve the cost reduction and minimum rejects, minimum reprocessing in manufacturing. We give higher level of assurance of the quality than traditional methods. Whole activity of development, scale up, even validation of the batches with the quality by design and commercializing is more transparent and predictable. There are minimum compliance issues because minimum failures, minimum reprocessing. And as such, your regulatory agencies are always happy with such organizations who are having as good as nil failures and minimum failures, so that they are satisfied with their during their inspections. And there is a high image and credibility because we are having continuous improvement on the quality of the product. Thus, we achieve right first time in the organization. Right first time is the goal, so that minimum failures. We are having robust development, improved development capabilities and flexibility of the process to minimize the quality issues. Thus, we are achieving zero defect of every batch, every unit of the batch, every day, every month, every year after year. Our productivity increases because the time of rejection is minimized, and that is used to manufacture the new batches. Science in risk-based understanding has enhanced, and we are satisfying the regulatory agencies and thus the patients and the customers in the market. This is where I was talking to you. What is PAT? It is helping us the control strategy to improve the productivity, to improve the quality, to lower the risk of the quality of the product, and automatically as your quality improves, as consistency in manufacturing improves, you are able to give the product timely to the market, and automatically it helps the business of the organization. And that is why I always call process analytical technology as profit after tax to the organization. And this is the way we convince to the finance department to give the funds to invest into this technology. So profit after tax to achieve with the help of process analytical technology is a great thing to happen us. And thus, look at this slide. The productivity improves every day, every month, because your rejections are minimized, reworking is minimized, reprocessing is minimized, and the same time is used to take the new fresh batches to manufacture. And you see here, the quality person in traditional way is always under pressure. Now, with this concept, whole organization, including the quality person, are extremely happy as productivity goes up and up and up. When we talk of opportunities, there are also obviously there are challenges to the organization to achieve these opportunities. Then the biggest thing is to have a cultural change in the organization. The mindset of the organization has to be changed to have culture of quality in the organization, right from top to the bottom level of the organization. We have to keep in mind every person in the organization is equally important. There cannot be two standards or yardsticks for two different persons. Everybody is doing important work. I always, whenever I go to my assignments and consulting to the organization, I always tell to the board of directors that this quality culture has to start from the top managing director of the company or the owner of the company. They have to ensure that every person in the company only works for the quality of the product, only works for the patient's interest and nothing else. No compromises. And the quality culture every day in the manufacturing, testing, and before that in research and development is essential to implement. And it's a challenge. It takes time. Human mindset is very, very interesting, very funny. Some people get convinced, some people don't get convinced. Everybody has right to express. Every word, every communication has to be heard by the management and see how to convince them and what are the methods, what can be done better to convince them to start implementing quality culture in the organization. Knowledge management is another challenge which organization also faces. That is why continuous trainings are required, continuous refreshers are required, and this training and this knowledge management sharing is very, very important as we are doing today. And I appreciate your university for organizing this wonderful 3-day webinar series for the faculty members as well as for the students of the university and the colleges. This also helps to reduce the pressures on every department and it speeds up the operations to avoid the risk of failure. And to understand the risk and the remediation of that risk also is a big challenge in the organization. And to collect this data of the everyday results coming in and put it in a right perspective, rightly interpret it is also a challenge to the company. We have seen in last 8-9 years, India has suffered very badly, rather world, not only India, but whole world of pharmaceutical industry is a data integrity problem. Regulatory agencies in Europe, in United States, and even in India, they have observed few companies are manipulating the data. And with continuous warning letters issued to the companies, issuing non-compliance certificates from Europe to these companies since 2007-8 onwards. Now we are in a much better situation today in India. Let me assure you on that. Our companies have invested a lot to ensure the data integrity, compliance right from research and development itself till testing of the product and going to the market. Every stage, data integrity compliance is being addressed and it is being implemented with lot of investment and reviews and continuous audits, internal audits of the organization from this perspective, even having a very senior level compliance officer to ensure data integrity compliance is there every minute throughout the organization. For some, data integrity compliance in R&D is a big challenge today. Another challenge is the lack of technology and the knowledge to execute it. And the resources for that is also a challenge. Supplier variability quality is a big issue. As I told you initially, the garbage in, garbage out. So if you get garbage, you are going to have problem in your manufacturing, and the product also will be very bad. So this was also a challenge that is also getting overcome as the time passes. PAT implementation, process analytical implementation, is also a challenge, and there are knowledge of this process implementation. PAT implementation is essential to have a right people, to have a right understanding what should be the tools for the PAT implementation, what kind of analyzers I need in line or at line during monitoring the CPPs in manufacturing. And the skill requirement is very high, and slowly these things are also changing from the skill requirements, and lot of engineers are also and expert from the pharmacy education. I have been talking to the government agencies. I am on their committee to suggest the changes to them. Even I am regularly in touch with Mr. B.Suresh, Dr.B.Suresh, who is a PCI chairman, to suggest so many changes into the syllabus of the students. And so many suggestions have been given, and he is looking into it and very positively. And I appreciate Dr. Suresh intervention from that perspective. Another challenge which I would like to see to overcome very quickly now, also in India, manufacturing. And this continuous manufacturing process is all 24 hours you're manufacturing same product, same batch. Normally, traditionally, there are several activities as a batch manufacturing. Getting API from the market, then bringing it to our warehouse, testing it, releasing it, using it for manufacturing the product, tablet, capsule, etc. And again, taste the blend of the product, then compress the tablet, capsule. If capsule is there, make capsules. If tablet is there, make coating if required. And these several steps, it takes a lot of time. Whereas in continuous manufacturing, operations are in one machine. And what's that? This is called continuous manufacturing machine with several ultra reactors inside. I was extremely lucky to have met Dr. Alan Meyerson of Massachusetts Institute of Technology, who has process that you don't have to buy active drug substance from the market. You have to buy only intermediates. Convert them, these intermediate, in your factory to active drug substance, and immediately use that active drug substance into the tablet manufacturing. And this is called continuous manufacturing, ultra lean manufacturing. It's a radical transformation, revolutionizing the manufacturing activity in today's time. And this has been introduced initially by Novartis. And it starts with chemical synthesis inside and converting that chemical into the tablet manufacturing. And this is where it requires process analytical technology intervention. and which this PAT monitoring does the testing of all the process parameter inside, and then only you achieve the desired result. So the speed of manufacturing is very, very high in this area. So I was extremely lucky and happy that I could spend 3 hours with Dr. Alan Meyerson, and who gave me a lot of knowledge from this perspective. Then with this, so far on challenges, there are future challenges as well. There are so many technologies which are coming. And just now you heard before my talk, Dr. Amit Agarwal talking on telemedicine. There are artificial intelligence technologies. There is a Internet of Things, IoT, supply chain automation with the help of blockchain. So throughout the process now, we are moving into the area of automation, artificial intelligence. And this is also should be treated as a future challenges for the betterment of the processes in manufacturing in the interest of patients. So now today, some organizations globally are using AI technology in research and development as a product marker for development of the product, in manufacturing of the product, in quality of the product, quality assurance, quality control laboratories, supply chain. After this, in various activities, this AI and robotics people have started using already. India is comparatively behind in this area. But if you see this whole activity, what we are talking here is of different in manufacturing, testing, and supply chain. But now interesting part, just to give a glimpse of this AI, I would like to show you how it will be used in training also. Now the video I'm going to show you now is that the trainer is in Las Vegas with the audience in Las Vegas and talking in English. At the same time, this trainer is addressing the Japan media in Japanese language, and this trainer doesn't know Japan. All this is achieved because of AI technology. So let's see how it is happening, then we'll come back to this.
Now, it's a pleasure to be here in Las Vegas to present to you. Now, I get invited to do keynotes across the globe. And while it's easy for me to be here in Las Vegas, it isn't always easy for me to travel across the world. And even when I do, I can't always speak the local language. Well, what if neither language nor distance mattered for me to deliver a fantastic keynote? What if technology could help me be anywhere I needed to be and speak any language I wanted? We are bringing together the power of mixed reality and Azure AI services to create a truly game-changing experience. What you're about to see is an exact hologram of me wearing the same outfit that we recently captured at a mixed reality studio. And I don't speak Japanese, but what if I wanted to deliver my keynote in Japanese? Using Azure AI technology, I can translate my English into Japanese and train it to sound exactly like me, the same voice tones, those same inflections. Now we've brought this together, my hologram and Azure AI, to show you what's possible. So first, I'm going to put on my HoloLens 2 here, and then we'll flip in the room to the special camera so you can see exactly what I'm seeing. Let's get started. First, let me introduce you to Mini-Me. There she is, my perfect holograph. And thanks to the power of HoloLens 2, she just floats right with me. I'm literally holding my hologram so natural. Now she's a little small to do a keynote. So let's get her up so she can do full-size Japanese keynote. Render keynote.
ホログラムになることには本当に驚くべきことがあります。私たちは最新の複合現実キャプチャ技術を使用し、私のホログラムを作成しています。実在の人物をホログラムとして見たことがあるかもしれませんが、私が実際に日本語を話しているのが新しいのです。私は日本語を話しませんが、私の声とホログラムは完璧な日本語で話しています。これはニューラルテキスト読み上げと呼ばれる最新の人工知能技術、いわゆるニューラル TTS を使用しています。私たちは自分の声の録音を使用し、私のように聞こえる私自身の個人的な音声署名を作成します。日本語からフランス語、ポルトガル語まで話せます。今、この技術は私たちが働き、遊び、生活する方法に関して、世界中の国境や障壁を取り除くことができると想像してみてください。まさに SF が現実になるところです。
Now, as you can see, this is mind-blowing technology. And what you just saw was my life-size hologram, my exact replica, rendered here in real time, speaking Japanese with my unique voice signature. We use mixed reality technology to create my hologram and render it here live. Then we use Azure's speech-to-text capability and English transcription to get my speech, translate to get the speech into Japanese, and finally applied neural text-to-speech technology so it sounded exactly like me, just speaking Japanese. And the most amazing part, all of these technologies exist today. The future is here. Now, it's a pleasure to be.
So do you see the difference, what is happening in the world, my dear audience? And it is such a beautiful thing is going to happen. And I always suggest during my talks at various places that the universities need to move into these areas to include some of these automation technologies as a part of syllabus. Why we adopt these newer technologies? Because these are all robust processes. Risk to the patient is reduced, improves the patient benefits. Also cost is lower, and you are handling the variability effectively to minimize the variability. Minimum time to launch and improve the credibility of the organization. So overall, as a summary, we are talking of improving the credibility of the organization, and I'm sure you all will look from this perspective, hold this presentation for the benefit of the audience, the students, the faculties as such. And this is where I end my talk. I'm now stopping sharing the presentation and open for discussion further.
Thank you so much, sir.
I'm sure you people like this automation, what is going to happen in the industry now.
May I request Dr. Sushma to please conclude the lecture of Professor Bashikar?
Yes, Dr. Puja. Sir, it is a very wonderful talk, and sir, you have given a very deep insight on into this QBD and touched upon the various aspects of the QBD, starting from the Six Sigma to what are the challenges for the today's for the pharma industries and the educational institution. And sir, the Six Sigma concept now is the academic research people, they are also implementing, because if you are not implementing, the papers are not going to be accepted. So students, they have started from the lab, they have to inculcate this culture, because nowadays the US FDA has been accepted. So like you share the data integrity, and also the papers also needs how you can process for the formulation development and the analysis and all. So each and everybody now adopting, but this is a very wonderful talk, because we always academics and very far from the industry. So you have given a very true insight what we generally the industry people they want, and how we can proceed for the quality of the product. So sir, it is a very wonderful talk. But just can I ask one question, sir?
No, 1 minute before you ask me the question.
Okay, sir.
Is that I always love to talk to the universities and the students at their place, only because I can explain very well the gaps between the current scenario of the universities and what is expected by the industry. There is a very big gap.
Yes, sir.
And the faculties as well as the students must know this to bridge the gap.
Yes, sir. Because sir, always we listen whenever my students they are going to the industry, they always call, ma'am, it is very different. Ma'am, jo lab mein karte the, it is very far from that laboratory experiment. Although theory is same, but the implementation and the work of doing it is totally different. So they say that whatever we generally do in the lab, it is not going to be following pharma industry, not even a single 1%. So that is always be there, sir. We would love to discuss with you, sir, these issues. We can see the questions. Now the forum is open for the questions. Then later on, I will ask the question.
No problem.
Any questions from
Ma'am, did you get any
sir?
Also.
Hello.
Hello. Yeah. Thank you so much, Professor Bhashnikar and Dr. Sushma Talegaonkar for gracing this lecture was and the topics. Please.
Just one question. Sir, you have mentioned the design space, although we are using it, but can you explain in more detail? Because design expert, the regulation are regulatory agencies, they are not asking for design expert. So sir, can you explain this design expert with a small example? Like we generally maintain the CPP and CQA in this design space.
I will. First of all, it is design space.
Yeah.
Okay. And the design expert is a software. Name of the software.
Yes.
Okay. Design space, it includes the outer range of compliance of a CPP or CQA.
Yes.
Okay. Now, for example, let's talk of granulation. Mixer, where the RPM, revolutions per minute, should be 15 RPM. And when we say that my batch will be design space of 5 to 20 RPM. Understand this concept. The design space for revolution per minute is 5 to 20. And so that's a complete range I'm looking at. Where for normal operating range, I am writing in a document to manufacturing between 10 to 15. 10 RPM to 15 RPM. I am asking them, we prefer this to comply the process. Chance is goes out because of some reason, batch will never fail. If it becomes the 9 RPM or 16 RPM, batch will never fail because you are already within the approved design space. That's the explanation for that.
Yes.
And this applies for every process. And even I strongly recommend various organizations now, while they file the document, ANDA submission or NDA to US European authorities, you must give the justification why particular process is not CPP.
Okay.
If particular quality attribute is not CQA, why? That also you should mention. That gives a very good impression to the regulatory agencies review team that these scientists are having a very good knowledge in depth. And that convinces them much better way.
Okay, sir.
Did I answer your question?
Sir. Yes, sir.
Thank you.
Once again, I thank Professor Bharshikar and Dr. Sushma Gaonkar. Thank you so much. Now, we will, with your kind permission, we would like to proceed towards our next session for the day. Our speaker, Dr. Jayagopal Meher, is already with us. Dr. Jayagopal Meher is FL. Pardon, sir?
Can I leave the meeting? Please permit me.
Yes, sir. Yes, sir. Please. Thank you so much. Yes, ma'am.
Thank you, sir.
Thanks a lot to all of you, as well as to Dr. Gupta ji. Thank you very much.
Thanks to Dr. Gupta. Thanks, Dr. Puja.
Thank you, sir.
Thanks to Sushma ji also for extremely well monitoring whole session.
Okay, thank you so much, sir. Thank you, sir.
So, Dr. Jayagopal Meher, he is F.R.D. scientist at Dr. Reddy's Private Limited, Hyderabad. And to chair his session, we are delighted to have Dr. Suresh Thareja with us. He is associate professor at Central University of Punjab, Bathinda. Thank you so much, Dr. Thareja and Dr. Jayagopal, for sparing time for us. Thanks a lot. Now, I would invite Dr. Suresh Thareja to please introduce our speaker of the day, Dr. Jayagopal Meher. Over to Thareja, sir.
Thanks, ma'am. Thanks so much. So, first of all, I'm thankful to organizers, especially Dr. G.D. Gupta, sir, for such kind invitation for this national conference. So, I would like to first introduce speaker of the day, speaker of this session. It is Dr. Jayagopal Meher. He is from Dr. Reddy's laboratory. He's a formulation scientist and he's having more than 10 years of experience in the area of formulation. And he has done his doctorate from PhD from Center Drug Research Institute, Lucknow, while his master's as well as bachelor in pharmacy from Odisha. He is having very good, although he's in industry, but his research profile is excellent. You can see he's having a 19 research publication along with H index of 18. Other than this, he's the winner of this Best Innovative Ideathon Hackathon 2020, and he has also got other awards like Sport Award, Young Scientist Award, and other so many travel grants also. So now, I would like to invite Dr. Jayagopal Meher to share his talk. So Dr. Jayagopal, welcome here, and now dice is yours. Please share your views. Thank you, sir. Please.
Thank you, Dr. Suresh, and thank you to all of you for inviting me for this talk. Screen.
Sure, sir. Sure.
I hope my screen is visible to all of you.
Yes, sir. It is visible.
Okay. So once again, I thank you all for inviting me for this wonderful session. And today I will be discussing with differentiated pharmaceutical product development, the future of pharmaceutical sciences. Let me congratulate you all, especially the management, the director, ISFCP, organizers of ISFCON 2021 and Conference 2021, and Dr. Chawla, Dr. Vineet, and other faculty members, resource persons, delegates, participants, collaborators, as well as the invitees guests, students, researchers, as well as the supporting staff for this wonderful session. And I thought of bringing this image because while going through your website, I was a little bit fascinated about looking these figures. So this is really very tremendous efforts by ISF, and I congratulate you all for this particular achievement also. So it's a declaration that there is no conflict of interest for my today's talk with the organization that I have worked, whether it is a government organization or private organization, and whatever I will be presenting are in the public domain. And I have a publication number also from Dr. Reddy's laboratory. So I have, after a long thought, I have decided that let me divide my deliberation into three different sectors. That is the pharmaceutical market, pharmaceutical product differentiation, as well as the research that I have done during my PhD, during my project tenure, as well as whatever we are doing here. And by this way, I thought that I will be able to justify the theme of this conference, that is recent advances in pharmaceutical sciences vision 2030. And I have seen some of the lectures also. They are very nice, and even the previous lectures I joined in between, and it was really very nice to listen such eminent speakers. So let's start with the pharmaceutical market. I would like to let you know that see this figure, that the prescription drugs is expected to reach almost around 1.4 trillion US dollar. So look at this figure; it's a very huge figure. And despite the challenges like the pandemic or whatever other natural challenges or any kind of challenges, innovative and effective therapies are continuing to be the game changer, or I can say the growth factor for pharmaceutical industry. Another at the left hand side, I would like to show, if I can zoom it. Are you able to see the zoom image? Can anyone please confirm me?
Yes, sir. Yes, sir.
Yeah. Thanks, Vinit. So this is the worldwide prescription drug sales in 2020 and 2026. It's growing with a rate of 7.4 CAGR, and we have two different columns for here. There's the drivers as well as the brakes. So you can see oncology. This is the 21.7% of the proportion of the 2026 industry sales will be from the oncology therapy. Why I am showing all these things? Because we need to understand, if we want to have a vision for 2030, we need to understand the market. Because pharmaceutical sciences is not only academic science, or it's not only a conventional or basic science. It has got immense potential in the market scale. That 530 numbers of FDA NMEs are going to be approved. Those are the pipelines. And additional sales of 230 billion US dollar additional sales are there in 2026 from the biotechnological products. Those are in focus now, as well as 10.8% will be the projected values for the orphan drugs. At the same time, if you can see the breaks also, that 7.8 billion, there is a decline because of this COVID situation. A reduction in the 1.9% in the CAGR for this anti-rheumatic drugs. And further also, approximately 252 billion sales are at risk because of the patent expiry. This is a very important point, that patent expiries, that we people in the industry that look into this. So this is also important data that we should understand, that what exactly is the current market trend. And further, there is a 3.2% in the decrease in the spend in the industry, or the industrial R&D, I would like to say. So with these, I would just like to convey that this is the current trend, although it is dynamic in nature, but this is the current trend of the pharmaceutical market. Further, let me show you this image. Here you can see there are two graphs, two lines are there. So one is for the biotechnological. Let me use the pointer. Yeah. So this one is with the biotechnological drugs, and this one with each with the conventional drugs. So here you can see that initially the conventional drugs or the blockbuster drugs or the even NDDS also we can say, those were having a major sector for the pharmaceutical sector, and the biotechnological products or the products with the innovation, product with differentiation, I would like to say, this is the topic of my presentation. So they were having a less share, but you can see it is with the age or into 2019, it is changing drastically, and in 2026 again it is projected that it will overtake, means the biotechnological product, the innovative product, or I can say the product with some differentiation in their nature, in their types, will definitely overtake the conventional system. So with this background, now let me tell you that which are the major focus area in the current global scenario for the pharmaceutical sector, which area, because a lot of good research that we are doing in the academics as well as in the industry. So where the industries are moving and what should be the focus of the academia also, so that they can have some tangible outcome or the commercial impact of their innovation or of their research. So here you can see that rheumatoid arthritis is one of the major sectors, and even the antivirals, anti-diabetic, then sensory organs, vaccines. Vaccines are again coming up, especially with the COVID, and in future time to come also this will have some impact. I'll discuss this later. But as a whole, if you can see, then oncology is leading. So onco products, the products that we are using for the cancer therapy, those are leading, and because this is the most dreadful disease of this century, I would like to say. So this is the trend, and these are the major segments where one should invest his time, money, as well as interest. This is the worldwide total pharmaceutical R&D spend in 2012 to 2026. That although it is increasing, but up to 2019 there was a 4.1 CAGR, whereas from 2019 to 2026 there is a decline. Although it's a dynamic data, I would like to say, it may change with future time to come, but this is the current trend now. So this is being the overall pharmaceutical market potential or market trend, I would like to say, the major focus area that we should target and globally. But now with the current industrial context, Indian context, with respect to the industries, what is the current scenario I would like to tell you. So approximately 40% of the generic for the biggest market, that is the US market, that we are supplying. About 25% of the prescription drugs that were prescribed in the UK, we are also able to cater that. This is a very huge number. Global vaccine demand we are fulfilling, and with all these figures also, let us see the fourth point. The global formulation trade value is around 652 billion US dollar. That was in 2019 data. It somehow it is increased in 2021. India's share for the export for of the in the global trade was only 2.5%. So this is a very less number, right? And so what is the projection now? If we grow at a rate of CAGR of 12%, presently we are at around 6%, then we may reach to a 130 billion business by 2030 from a 41.7 billion in 2020. So this is the projection. But in order to achieve this, means just we have to double our potential, whether it's industry research, industry, academic collaboration and all. So we have to achieve this target, and this is our target by 2030. So in order to achieve this, what we exactly have to understand? So innovation-led research and development, we must understand. And we must understand the healthcare delivery. What is the current need of the healthcare delivery that we should focus on? Manufacturing and supply chain. Supply chain is very important, because if we are manufacturing, we are ideating, we are manufacturing, but we are unable to reach to the market, then also it's of no use. So this manufacturing and supply chain, it's very important. And to the market access, as I told now. So for this, we need to again focus on further. Those are the industry academic collaboration. This is well established in the Western culture. As in the previous presentation also, we have seen that how MIT professor, they have innovated something. that tablet manufacturing technique, and Novartis is following that. So many examples are there, starting from the gene therapy and all. So from academia, knowledge transfer, that is the KT, from the academia to industry, it flows well in the Western culture, and we are now in a right situation, or the industrial revolution, fourth industrial revolution, I would like to say. We must focus on this particular thing. Furthermore, increase in the private investment. What I mean to say is that any academic research is being well funded by the government organizations, government agencies, especially in a country like India. But again, the private investment for a much higher risk development, and to establish a strong innovation ecosystem, because innovation will never happen unless until there is an ecosystem for that. And even the investment, the private investment, and even the collaboration between the academia and the industry, that is more important for this one. And all these things now, gone those days that we were just focusing on a blockbuster drug, or we were just focusing on a drug discovery, but now everything is centric, quite targeted toward the patient. That's why we call it as a patient-centric feature or patient-centric innovation. So with this brief background of the market, now let me tell something about the pharmaceutical product differentiation. What differentiation we are trying to make in the pharmaceutical products? So differentiation is a core business strategy, because as I already mentioned that pharmaceuticals are more into research as well as business. So they have a commercial, significant commercial impact. So it's a core business strategy. And here what we exactly want to do, we have to understand, or we have to deliver something that is distinctive in nature. Distinctive features we have to identify, and we have to introduce some unique features to the product, as well as to ensure that some unique USP is there with the product, so that it can have some importance in the market. This is without compromising the safety and efficacy of the product. So apart from the safety and efficacy, that is not enough now. Apart from that, what differentiation we're trying to product development, that is more important now. So that differentiation strategy may be now in new dosage form, or new indication, or a different kind of indication, drug combination product. So this is the basic understanding of the pharmaceutical product differentiation, and this can be achieved by integration of the three different I would like to say aspects. Those are the products, because product is at the center. The services, because now this is not only the product but also the services, like the different AI, ML, and this internet on things. Many things are going on. These blockchain, many things are going on. So not only with the product, they will also cater the services like reminder, or like the compilation of the data, or reaching to the doctor online, and all these things. So these are coming under the services, and this can be achieved through the data and the technology. So integration of all these three, the product, services, data, and technology, this is going to be the pharmaceutical product differentiation in near time to come. So if you can see the traditional biopharma, they were having a till now they were having a good setup, and they were running their business with the generic products or the ANDA products. But now we are changing, and what are the changes? That we will now more focus on the prevention and early detection of the problem, because many of the things like cancer in a country like India, it is detected in a very last stage. As you all know, that it is detected in a very last stage. So that stage, it is very difficult to cure the patient. So this is one of the important aspect, and we have to understand the prevention and the early detection of the disease, and that can be even achieved by usage of this data and technology. Further, we have to go for the customized treatment. Now it is not like that for every sort of disease we have a unique, very single type of approach, or I can say a very traditional type of approach. Now we have to have a customized approach for the treatment. And the third one is the curative therapy. Previously, allopathic medicines, they were being treated as a treatment of the symptoms. But now, because of this cell therapy as well as this genetic therapy, we are again moving towards the curative therapy. Then further, using the power of digital therapeutics, the power of data that we have seen, and precision intervention also means whatever we are doing with a very precise way. So those things we can, those things are now the new trend in the pharmaceutical product differentiation. But different diseases will be again impacted by this disrupted forces. And as you can see that if we can categorize these diseases like the genetic diseases and the complex multifactorial diseases, then we have a lot of number of diseases starting from the Parkinson disease and the Alzheimer, the diabetes, the hypertension, cancer and all. And these can be treated by different approaches like the genetic treatment and all that can be done by introducing the cellular and genetic therapy. That will be the curative therapy that I was talking about. And the complex or multi-factorial diseases that can be treated by this custom treatment therapy. So this is now the new trend. So pharmaceutical product differentiation. So if you want to have a pharmaceutical product differentiation, then what exactly we have to keep into our mind? So first of all, we have to raise the bar. Means we have to be classed apart from whatever there is in the market, so that we can have a differentiation in our product. And we have to solve a problem in a different way, or we have to solve a different problem. Suppose a particular drug is there and it is having a side effect or something. Then if we are able to achieve that, then this is a significant improvement on the differentiation. And further precision play that as I already told, that this will be a patient-centric, completely patient-centric, and how this treatment history and this patient characteristic, its behavior, as well as its genetic profile and all, how they are playing the role very precisely. We have to understand that, and by that way we can have a differentiated treatment for this diseases also, and further also build a different yardstick. And we have to focus here on the new measure of success, that is unmet and the unmet need of the patient. So with this background, what exactly is the market and what exactly is the differentiation in the product development, I thought of discussing with you guys that what the journey, how it is relevant to the industry. So it will be again more important for more justifiable for this particular talk, I think, right? So during my tenure, I worked with the CSIR CMAP as a research fellow. Then CSIR CDRA also I worked. Presently, I'm working in Dr.'s laboratory.
Sir, I think there is some network issue.
Yes.
Many of the essential oils. We thought of.
Sorry to interrupt you, sir. Sir, I think you lost your connection in between.
I think he got disconnected now.
How are you, sir? Yes, sir.
Yeah, I'm fine.
काफी संसद से आपके बारे में सुना, बातचीत हुई कभी कई बार.
Okay, okay.
आज मिलने का अवसर मौका मिला फेस टू फेस सर.
So you are in which department?
Pharmaceutics.
Okay, okay. That's great. I think someone can contact him.
means from there you lost the connection, sir.
Okay, okay, Vinith. So, do anyone remember? Then, in just a minute.
You just started your our R&D work.
From CSR,
from your R&D work.
Okay.
Yeah, please.
Okay. I'm sorry, I don't know how it happened, but I was just looking at to the screen, to the presentation. So probably that was the reason. Are you able to see my screen now?
Yes, sir.
Yes, yes.
Yes. I think I was here, right?
Yes, yes. You just started this slide. Yes.
Okay. So I was talking to you guys that with this brief introduction of this, what is the industrial prospect, what is the market prospect, and what is the product differentiation aspects, and what is the need, which are the areas that we should focus, and what exactly the product differentiation that we can create. I thought of integrating whatever the academic, whatever the scientific research that we did, so that you can have some understanding that how this is also relevant to the thing that we are doing in the industry, because this is again a matter of question that whatever we are doing in the academia or in the research labs, whether they are of use or they are of no use in the industry. Definitely they are of use, but we need to understand its importance as well as its timing as well as the market potential. Just moving toward that, I worked in 3 different organizations till now, and this is CSR CMAP, CSR CDRI, and Dr.B.Suresh laboratory. So in CMAP, we were trying to develop some of the topical formulation, and there we faced a lot of challenges with respect to incorporating the oils, that is the essential oils. They're therapeutically tremendous, but in order to convert them into a tangible product, a stable product, it was a very typical task for us, because we were facing a lot of issues with the stability and all. So that helped us to understand deep into the science that what exactly is happening, and from there we determined the hydrophilic lipophilic balance of those oils, and that again helped us how to develop a stable formulation. And those hydrophilic lipophilic balance of these products were not available in the literature. We started determining that, and it again helped us for a development of a stable product. But in due course of time, again another thing came. So this HLB determination, I would like to say, this was not patient centric, but this is the formulation centric approach, that how a formulator can be benefited by deep diving into the science and to understand the real problem. But in due course of time, again we whether it's a RX product or whether it is a cosmeceutical or whether it is a cosmetic product, human perception is associated with the semi-solid products or topical products. So there we try to understand the physico-perceptual parameter of the products. This term is also probably should be new to my audience. And physico-chemical parameters are there, and physico-mechanical you can say, but physico-perceptual parameter. So based on the physico-chemical properties of the product, how a human perception is being changed. That is that was the approach. And due to lack of time, I will not be able to go in deep into that. But I request you to go through these links that I have provided, and you can have a good journey of the product development, especially to the pharmaceutical formulation. So with this understanding of this hydrophilic lipophilic balance and all, now I got an opportunity to work with pharmaceutical division CDRI. So there, when I went again, because oncology is the main therapeutic area that which many people are working now. So we thought of working in that particular area. We published a couple of papers also, and at that time, again, the patient-centric approach came to our mind, that whatever the treatment for breast cancer is available, so how can we have a robust formulation for that one, and again a patient-centric formulation. Cancer, it's a very dreadful disease, and paclitaxel is one of the drugs that is being used for this disease. It's a very classical drug, and clinically it is available as Taxol, but Taxol is having some issues like the hypersensitivity and leaching, because it is an intravenous formulation. It is given through this bags, so leaching is one of the issue, and That's why these days leachable and extractables are one of the main thing that we study, whether it is a liquid, whether it is a semi-solid product also. We have to submit all these things to the regulatory agency like USFDA. Again, the issues with this Taxol is again co-administration of other drugs in order to reduce the side effect and IV route. So these were the major problems. So a wild thought came to the mind that whether are we able to have a oral formulation for this paclitaxel. Although it was wild because looking into this physicochemical properties of this drug, it's very difficult or it's very typical to develop a oral formulation of paclitaxel, but still we tried. And we were motivated from some of the existing formulation like the cyclosporine. It is having a similar kind of behavior or physicochemical properties to that of paclitaxel. And again, this turmeric milk also, it is also turmeric is very hydrophobic in nature. But while it is given with milk, its bioavailability enhanced. Cholesterol, you can see that more than 50% is absorbed when we take orally. Whereas if you can see the log P is 12 and the solubility is 10 nanogram per ml. So all these things again motivated us that yes, we have a way, just we have to go. So with this, we started investigating the oral formulation of paclitaxel, and we have developed this is the trend that we have to use this QBT, and we have to use we have to understand the design space. We have to use different formulation combination. So we have used different experimental design techniques and developed the formulation that is a solid acidities formulation. For here, we have taken vitamin E and TPGS that we have integrated with our anti-cancer formulation and other surfactant, co-surfactant like your capriol and labrafec. This acidities product, we have with the biological parameters or the cellular parameters. That was more interesting because unless and until we have some effect. So this is all about the formulation development part. I'm not going in details because a lot more I have to cover. And this is the in vitro pharmaceutical characterization, but that we determine the size of the particle as well as its release characteristic, its distribution pattern, how it is behaving. And after getting a robust formulation, this was a major chunk of our activities. After developing a good formulation, then we have gone for this stability studies as well as this cytotoxicity and cellular optic studies. So here you can see we have evaluated it against two different breast cancer cell line, that is MCF7 as well as MDMB231, and here you can see this SIPTX acidities, that is the solid paclitaxel acidities formulation, Taxol as well as the blank formulation. So here you can see at 72-hour point of time that we are having a good cytotoxic effect of this particular drug. And in this MDMB231 cell lines also, we were getting a very good response for this acidities formulation. When we compared this, this is all about this cytotoxic MTTSA. And when we compared with our formulation with the blank, then here you can see that this formulation is being uptake by this cellular cells, the breast cancer cells, in a very good way. And that we have seen. So now we got a clear indication that our formulation is stable pharmaceutically, and it is having some cytotoxic effect. But now, if it is having some cytotoxic effect, then what exactly is happening to the cells? So for that, we have gone for this cellular and nuclear morphology of this product. So let me just zoom in. I don't think that I can show you a very good kind of image because this is again copied from somewhere from my publication. So here you can see, at least in the number you can see. So this is the control treatment. This is the blank formulation. This is the Taxol formulation, and this is the formulation that we have developed. So here you can see in the control at 72 hours, the cells are proliferating because cancer cells, they tend to proliferate very fast. And here you can see with this blank formulation, with this Taxol formulation, with this novel formulation that we developed, it is showing somehow reduction in the cell population. Now, this is the DAPI staining. So up to now we can understand this. Yes, the cell is cytotoxic in nature and the cells number is also reducing. But what exactly is happening to the cells, whether this is necrosis, whether this is apoptosis, or what exactly is happening, in order to have some understanding on that, we have done this DAPI staining. So here you can see these are the very intact cell, the nuclear. So DAPI stains the nuclear content of the drug, of the cells. So here you can see that they are quite intact cells, quite healthy cells. But here you can see they are fragmented. So nuclear fragmentation has happened, and those nuclear fragments are being stained by this DAPI. So here, from here, we can understand that yes, the drug is having some cytotoxic effect. Further, we move ahead, and these are the in vitro characterization that I'm trying to show you. And this is done in CDRI in collaboration with our endocrinology division. And this similar kind of approach we have in the industry also. Suppose we have some potential molecule, and if it is a new molecule, if you're going for a generic development, then no use of doing all these things. But if you're going for a innovative one, if you're going for a new one, and if you're going for a differentiated one, then this path we have to follow. And I hope then this will be definitely useful for you guys. Experiment that we did. Here you can see this is the control, and this is at 42 hours. So here the cells were migrating towards each other. They have the tendency to migrate towards each other. And this is the blank formulation. This is again the Taxol, and this is the acid formulation. So here you can see the gap is more here, and the gap is less here. It means the cells are unable to migrate because we are restricting the cells. We are killing the cells. That's why they are unable to migrate from one place to another place. Up till now, we have a qualitative understanding that what exactly is happening. Our formulation is, whether it is effective or not, that we have seen. Further, we move ahead with the cell cycle distribution, that how this, in which particular, because we have a cell cycle, right? We have different phases. So at which particular stage this formulation is blocking this cell or arresting the cell growth, cell cycle movement. So here we can see that G2M phase arrest we have seen, and this is significant in case of both in case of Taxol as well as our formulation. Again, these are some of the other studies like the apoptosis. Apoptosis is a programmed cell death. As we're talking about a lot of program, this AI and ML, but supernaturally our body is also like a program. And apoptosis, many programs are happening in our body simultaneously, as you all know. So apoptosis is also a very beautiful program, very systematic program. If it happens, then it is desirable in case of, especially in case of cancer. So here we wanted to understand that whether our formulation is doing some apoptosis or it is showing some other kind of cell death, like necrosis and all. If necrosis is not desirable, or even necroptosis is also another type of cell death that may also happen with the formulation, different formulation. So here you can see that this is the treatment group, and here in this particular quadrant, which is showing the cell to move towards the apoptosis, it is significant in case of the acid formulation. And with the Taxol also it is moving out, but whereas in the control you cannot see the cell are not moving towards the apoptosis. Membrane potential also we try to understand. Apoptosis is happening, but again it is through the intrinsic or extrinsic pathway that we try to understand. And through this mitochondrial membrane potential, we understood that this is happening towards the intrinsic pathway. It's following an intrinsic pathway, and further cellular cascade is happening, or which are the genes those are responsible for this particular kind of apoptosis. That also we have studied, but I think I'll not be able to present all those things because of shortage of time. Now this reactive species generation, the ROS generation, that this is another indication that how the cells respond to the anti-cancer formulation, and if they are increasing the ROS level, then also there is a chances that the cell will die fast. With this in vitro studies, the qualitative studies, the cytotoxicity study, the morphological studies, the wound healing studies, and further this specific in vitro studies like the apoptosis and cell cycle distribution, like this mitochondrial membrane potential and ROS generation, we understood that yes, the formulation is potential enough to, and it is a different kind of formulation that we are approaching towards a what we have hypothesized that whether this oral formulation will work or not. So up to in vitro level, we thought we could find that this is working well. Now the main study started. The most interesting study started. That up till now we have a good result, but let us understand this bioavailability aspect of this particular dosage form. If the product is not bioavailable, then whatever we have done, that cannot be taken further. So in the bioavailability study, we have given, as you can see, this different groups, Taxol and PTX SADDS, and this Taxol as a IV administration, 4 animals per group we have taken, and we have studied it in rabbit. So here you can see this bioavailability is approximately 18.6 here, whereas in the Taxol it is 4.75. So approximately there is a 4-fold increase in the bioavailability, I would like to say, because of this SADDS, paclitaxel SADDS. We also investigated that what exactly, because Taxol is not having any proper bioavailability, because it's a simple admixture with the chromophore EL and all. But our formulation, it's a emulsified system, and it is having some specific mechanism by which it is going towards the cells, or it is penetrating through the GIT. Those study we have done, all those things. So after this bioavailability study, we were fortunate enough that we had some cancer models available in the other department, like the anti-cancer department. I worked with Dr. Shivani Dikshit and Dr. Sekia and all, Dr. Datta. So they were having their, because they are into the drug discovery line, and they have studied the anti-cancer efficacy of various plant extracts or various new synthesized molecule, in-house synthesized molecule. So with their collaboration, we have studied this one, this in vivo efficacy study. So here we have done this with the HD rat, that is the Spordelli rat. See, this is the campaign that we have done, and it was a 21 days treatment. So we have treated the animals through oral route, the vehicle, the Taxol, the blank, as well as this double formulation, and we have evaluated them with respect to the body weight, tumor volume, tumor weight, survival, organ index, histopathology, apoptosis, as well as lung metastasis. I hope I am clear till now.
Yes.
Yeah. Okay.
Please
Thanks.
go ahead.
Thanks.
Yep.
Thanks. Yeah. If any point of time anyone needs any clarification, please don't hesitate to stop me. Yeah. So with this treatment group, you can see some of the images. This is the first image shows the inoculation of the cells. So what exactly we do? We grow the cells in the laboratory, and once the cells are grown in a LS7 cells we have used for this particular study, and once the cells are grown in a particular population, then we take those cells and we inject those cells into the mammary fat pad of the rats. So these are the female rats, and this particular area is the mammary fat pad area. So here we have inoculated the cells, and after a time period of around 2 to 3 weeks, we generally see the tumor that is in a very small tumor is developed, and with due time it again develops, and lot of mortality also happens because of this again getting a consideration, this can be measured with this electronic slide caliper or Venn caliper, that through the oral garbage how we are dosing the rats, and this is the on the day of sacrifice, this is the tumor that we are able to see, and this is the excised tumor, and this is the way that we have removed the tumors from the rat on the day of sacrifice. So here you can see the body weight. We have investigated that what exactly is happening to the body weight. So you can see that in case of treatment group, the body weight of the rats are decreasing because they develop the tendency of diarrhea and not eating the food. So those tendencies they develop because of this chemotherapeutic treatment. So here we have seen the reduction in the weight of these animals. And further, on the day of sacrifice, that is after 21 days of the treatment, we have removed the tumors. Here you can see these are the tumors, and these are the different treatment groups. This is the control group. This is the blank Taxol group, and this is the blank acidides. Here you can correlate. So the first one is the vehicle, and the second one is the Taxol, and the third one is the blank formulation, and the fourth one is the that formulation that we have prepared, we have developed. So here you can see clearly that there is a significant reduction in the tumor volume as well as tumor weight. Here we have some of the tumor weight of this treatment group. So now we have done the survival analysis also, and we could find that Taxol treatment versus this vehicle control. So in the vehicle control team, they were dying, or we can say they could not survive because of the tumor load. And with the treatment group, the number of animals were comparatively more in comparison to the vehicle control or the blank treatment. And with this organ index also we have seen, because this anti-tumor or cytotoxic drugs, they have some of the impact. They have some significant impact on the organs like the heart, liver, kidney, lungs, and spleen. But we could not see any serious damage because it was a study for a particular period of time. So we could not see a serious impact on the organs. Liver, we have seen in some cases we have seen there are some of the issues, and this is with respect to weight. So again, with respect to the metastasis, that again we have seen that breast, this is very near to the lung area. So the nearest metastasis is happening to the lung, and that we could see in the control group, whereas in this treatment group we could not see, because somehow we are able to stop the growth of these cells in the treatment group. This is apoptosis in the tumor tissue also. Previously, we have seen the apoptosis in the cells, that is the breast cancer cells, but now this is excised tumor, and with this tumor also we could see that how this is behaving, and you can see that with this treatment group, there is apoptosis in the cells. And tumor histopathology, again, here you see the density of the cells, because as we all know that cancer means unlimited growth. So here you can see the number of cells is very dense. It is very dense packed cells, I can say, in the control treatment. In the blank also almost similar kind of things that we can see. And in the Taxol treatment and in the acidities treatment, that we could see that yes, there is some reduction in the tumor burden. So further, as I was saying to you that this is the control group, and you can see that here we got some of the lung metastasis. So lung nodules we can see here, the white patches. This is not clear in the image, but here you can see that this is the control treatment, and the cells are again, the lung is also affected, and now it is again cancerous. And whereas in the acidities treated group, that you can see any kind of, although in some animals it was there, but significantly we could not see the metastasis in case of the treatment group. This is again acute toxicity studies, that as I was saying you in the organ index, that acute toxicity also we want to understand. So with the stomach, because you were administering this cytotoxic drug to the oral cavity, and through the oral cavity to the stomach. So there was some disruption in the stomach lining and all. That is again the feature scope of this work. And but in the other organs like heart and kidney and lung and liver or spleen, so in the treatment group we could not see any significant impact of the cytotoxic drug with respect to any damage to the. So with this we can I can conclude here that with a patient-centric need. So here the concept, whatever hypothesis we have tested, and we are further moving with this, that whether paclitaxel is being able to being delivered through this oral route, because PGP efflux and all, everything, so many things are there. That's again a part of my PhD research, that what exactly is the mechanism, that how it is happening. And very short I can say that survivin is one of the marker that we have studied. So So those things, as well as this, how it is impacting, or how it is penetrating to the gastrointestinal tract, all those things we have studied, and that can be discussed in any another forum. And because I thought that being in time is important, being in industry, so 45 minutes I thought of concluding all these things, major study. And with this, I can say that patient-centric innovation we tried to do, and it is on a process. And in a industry also, we are more focused into this kind of innovation that can have some market disruption. So with this, I would like to say that patient-centric innovation through pharmaceutical product differentiation. This is a differentiated product because it is available in the IV route or the parenteral route, but if you're trying to get a oral formulation for this particular thing, then this is again a differentiation. And this you can achieve by any means, by the use of power, by the use of technology, by the use of data or whatever. So those things may come and may go, but this patient-centric innovation through pharmaceutical product development will remain same. And I hope that I have justified the theme of this conference. And I would like to stop here. And I thanks once again all of you, the organizers and the directors, sir, and the faculty members, the coordinator, and for this opportunity. I'm really happy, and thanks once again.
Yes, thank you, Dr. J, for beautifully explaining. Means your presentation was totally as per the theme of the conference. It was like Vision 2030, and you have beautifully divided your this presentation into 3 sections. The first one was very well explained, the pharmaceutical market. You explained the you can say about the oncology and the biotechnology product that will going to change the dimension of the pharmaceutical market. And your second section, that was again, anyone who is working in the area of this R&D, this product differentiation, this is very, very important. You have beautifully explained the means there is a need for this industry and academia collaboration. So it means even government is giving emphasis on this thing. It means those industry and academia should combine, and simultaneously you have included one important point, that is private investment, because without that ecosystem, if you will think about the product, discovering a drug from the beginning to end, you require that thing. Means if you will think, say, means the academia alone can develop a molecule, it's not feasible. So that collaboration is very, very important. Simultaneously, that ecosystem which we lack currently in India, that is also very important. Then you have beautifully explained your research also. You have given an example of Facilitexal, a real-time problem. You have you have worked in the area CSI labs as well as in Dr. Reddy's lab. So it was a really nice presentation covering future of so pharmaceutical sciences and what we will be in you can say 2030. So you would beautifully explain many things. And I would like to add, means like you people, you are sitting in the pharmaceutical industry, and we people, we are in academia. You should give means you can assign it's not a means paid job also. What I will say, you can assign some problems to us, because those PG project whatsoever we are guiding, or those PhD things, those are those conventional problems only. Like the real-time problem, because you are sitting in that chair, and you are working in those labs, the real-time problem. If you will going to assign those real-time problems, then we will come out with the solution, and then we can submit the solution to you. If it is means appreciable, you people will going to take it anyway, right? Because it is your problem, and we are trying to find out some answer to your problem. So rather than doing that conventional problem, like you are saying, patient-centric thing, you are focusing, you are innovation should be patient-centric. Now, what I will say about our research dimension whatsoever, it should be in academia, it should be industry-centric. Whatsoever problem you people will going to give to us, we're going to find out solution. In this way, we can create the ecosystem, because you will going to provide us that investment thing which we lack currently. It's a major problem which we people are lacking in academia.
Right.
If you will say, government will going to provide you everything, it's not feasible. So we will going to stand. We will think about this thing. Means after 2030, we will going to stand at the same position if we will not going to change our mindset. It is all about mindset.
Right.
So this is what means my idea. Like if you're going to share the problems, like we can create some platform type of thing where you people can come and you can like I am from Center University. This ISFCON is premier institute. You can say in the notes, and the autonomous institute. What else you require? And they are having that state of art facilities also, and they are open for this thing. I mean, so they will going to take it open-handedly if you're going to share those problems which you people are working upon. So that will be really, really great concept. Means combining the industry and academia. You don't require that. I will say for the financial thing also, because whatsoever project we are doing is it's like those conventional things. We are taking those formulation and doing those things, and it is just like only for award of a degree. So we have to change our mindset now. And it is we can't alone means I worked in a Novartis, you were talking about the blockbuster drug. So when I started working in the Novartis, but like their projection was like every 5 years they will going to discover 5 blockbuster molecules, because they were innovative. They were not like generic drug company. They were innovative, and what their aim was means yes, we will going to produce something innovative, because once a molecule, like you were saying in your presentation, there will be loss of $252 billion, market will go down. Exact quantum I'm telling you, because expiry of these patents. So they were aware about this thing. Means what happens to Pfizer after the expiry of Lipitor. So. So market goes down like anything if your patent will going to expire. So you require a replacement molecule. So this is what the thing, what we people, what we want, our academia wants. We want you guidance from you. For if the industry will going to assign a problem, we're going to find out the answer. If we're going to find out the answer, yes, you will going to provide us that ecosystem so that we can work in that direction also. So this way, I guess we can cover, and that should be the vision 2030. So thank you again, Dr. Jay, for sharing your views. If anyone want to ask any question, please you can ask. Please.
So Dr. Suresh, to your point, I would like to mention that you raised really a very good point, and I'm happy to know that you have some industrial experience also. So whatever the academic research that we do, so it's a trend like whether hypothesis testing, right? So so whether this is working or this is not working, that we usually want to understand.
Okay.
But the real challenge is like if you understand that impurity issue. Right. So nobody bothers about the impurities in the academia. Whether the API source is, what is the API source, what are the impurity profiling, what is happening to the impurities. Because if you're exceeding the impurities, no agency is going to accept you.
We will going to accept.
So that is one point that I would like to suggest, that develop some because you have time in industry. That is a problem, that you have stipulated period of time. So with this time frame, FY21, you have to work this. If you are not able to work, then it's gone. There is no market potential. So in that aspect, I would like to say that impurity profiling, understanding the impurities, and how to see, like as we work, that essential oil, we are unable to keep it inside the cream formulation, HLB system. Now, if we know the HLB, hydrophilic lipophilic balance, then we're comparatively, it's very easy for us to develop a cream formulation. In a similar way, suppose with the increasing temperature, with the increasing age, the impurity profile is increasing. So what are the measures that can be done? So you can assign a very specific type of project to your students, that suppose these are the molecules and they are having this impurity profiling, and now it's our duty to make a robot formulation with a very stipulated impurity profiling. So here we can minimize the gap. This is one thing. And another thing is that very often, suppose as per the guidance, that we need to go for this sameness, right? This Q3 sameness, qualitative, quantitative sameness, and this physicochemical sameness. And there we used to struggle a little bit with this IVRT and IVPT, if I'm talking about this topical formulation. So this is again a very good sector, I would like to say, that invest your time, energy and everything with this IVRT and IVPT techniques, so that, because you know that we are approaching the other industries, even the other CROs abroad, and they are charging a lot of money.
Yes.
Right. So if people in the industries, they can work on this two particular things. Two clues I am giving you. This first one is impurity profiling. Then second one is this Q3 sameness with respect to, especially with respect to this IIT and IBPT. Then it will be a great deal. And the third one again, any kind of innovation, because innovation is not confined to either industry or academia or anything. Any disruptive, then although definitely try to have industry collaboration with your institution. If it is possible, then we'll sit together and we'll try to understand that what exactly the innovative things that you have done, and that can be translated in the industrial setup. Even you see this 2DG, this is the innovation by this DRDO, right? And
Yes.
Yes, and our organization, that we took this manufacturing and all, and now this will be this is launched in the market.
Yes.
So that kind of thing, if it is innovation from ISF, if it is innovation from NIPER, or if it is innovation from CSIR, it doesn't matter. If it is innovation, if it is disruptive to the market, then definitely anyone can go and grab that opportunity. Yeah.
That's really great, Dr. J. So I will try to stay in touch with you so that we can have some that type of fruitful collaboration. So thank you so much, Dr. J. Thanks to organizers also, and thanks to special thanks due to Director Professor G.D. Gupta sir. Thank you so much, sir, for your kind.
Thank you, Dr. Thareja ji.
Thank you, sir.
Thank you, Dr. Jay.
Thanks a lot, Dr. Gupta.
Thanks and welcome. Thank you. Yes, continue, please, Thareja ji.
No, thank you so much, sir. So it's over from my side.
I would like to thank Dr. Jay Meher and Dr. Suresh Thareja for being here. And sir, your lecture was really very, very informative, and it was a very nice and healthy discussion too. So once again, on behalf of entire ISFCP family, I like to thank both of you that you have spared time from your busy schedule, sir. Thank you so much, sir. Thanks a lot. So Dr. Vineet wants to say something. Yeah,
please.
Yeah.
Thank you, sir. It was really very nice presentation, and it was like I'm learning again from you, and I've learned a lot from since I was there in CMA with you. So it was really nice, and thank you for accepting our invitation, sir. Thank you so much, sir.
Thanks a lot. Thanks. Yeah.
So, sir, should we proceed for the next session, Principal sir?
Bye.
Yeah. Our next speaker for this session has already joined us, and it's really my privilege to introduce him. Dr. G.D. Gupta, sir. I think I should continue. So it's a really nice of Dr. Jayant Karajgi for joining us today, and it's really a privilege for me to introduce such a renowned personality. Dr. Jayant Karajgi is a proud pharmacist by education as well as by training. This is the code that is set by Dr. Karajgi himself. His coming to his educational qualification, he is M.Pharm in pharmaceutics, NDDS, and PhD in pharmaceutical technology. Again, NDDS. MBA, he has done MBA through distance learning, and university gold medalist in B.Pharm as well as M.Pharm. Coming to his professional experience, it's a huge experience, but I would like to just compile a few, things from his career. Started his career in industry, research and development, formulation development at Torrent Research Center, Ahmedabad. Then he moved on to Ranbaxy Research Laboratories, Gurgaon, and followed by Sun Pharma, Mumbai, as a manager, formulation development. Then he moved to USA and worked in US generic industries for almost 7 years. Rose to the position of director, product development and technical tech services. He has cultivated knowledge of all the aspects of pharma industrial research, like analytical, bioequivalence, clinical, documentation, intellectual property, production, regulatory, etc. Then he moved back to India, and for the past 14 years, he have been serving at leadership positions as president, CSO, COO of reputed organizations like Milan, Aurobindo, Vocard, etc. Currently, he is serving as COO of Shilpa Medicare, a company which specializes in cancer care. During a career span of 29 years, he has launched more than 250 products in US and European market and many in Indian market, holder of more than 15 patents and many publications. Invited as speaker in reputed symposia, conferences and academic institutes across India. He is responsible for managing the entire business chain from product identification to product development to filing to approval to commercialization to supply chain to post-marketing activities and all business functions of the company. So I wholeheartedly, once again, with folded hands, I welcome Dr. Jayant Karajgi, and I'm really thankful for him to accept our invitation. Now, without taking much time, over to Karajgi sir directly. Over to you, sir.
Good afternoon, and thank you all for the introduction. Are you people able to hear me?
Yes, sir. Yes,
welcome, sir. Welcome, Dr. Karajgi, sir.
Good afternoon, Dr. Gupta. How are you?
Sir, very fine.
Okay. Let me get started. I will just share the file, the presentation.
वो स्क्रीन मूव नहीं कर रहा सर, जैसा है वैसा ऑलरेडी प्रेजेंटिंग हो रहा है, पर आप नहीं देख सकते, उनको स्क्रीन दिख रहा है। ये मैसेजेस क्यों आ रहे हैं? ये वो प्रेजेंट हो रहा है स्क्रीन में। कुछ नहीं सर।
कुर्सी पे आ जाओ, मुझे नहीं बैठना, बहुत देर से बैठी हूँ।
पूजा मैडम म्यूट कर लीजिए प्लीज।
Okay, can I start?
Dr. Sahab, you can just give in the mode of PPT. This is a full presentation mode.
Sure, I will do it. Is that okay?
Okay. Now, okay, perfect.
Okay, great. So good afternoon, everyone. Today, what I will try to do is, I will try to talk about certain things, which are otherwise not available in textbooks, and which are not available in published literature. So I will try to talk about things which are not available in books and which are not given and available in printed literature. I will try to talk more based on my last 29 years of experience in the pharmaceutical industry. Okay. I will start by telling you about how the pharmaceutical industry in general works. Okay. So whenever we start developing a new product or we want to develop a new product, which is what the pharma industry is all about, the first step which we have to do is to think. I think there is some background noise.
Sir, please continue now. Yes, sir.
So the first step that we have to do is to think, because without thinking, we cannot act. And for our actions to be correct, first our thinking has to be correct. So the first step when we talk about a new product development process in the pharma industry is called as ideation. Right? Now, ideas can come through a variety of means. Ideas can come through different means. We all know about serendipity. Although we keep on hearing this term and we think that it happens by accident, that sometimes somebody is doing something with some intention and a new thing comes out of it, which is what is called serendipity. But believe me, in actual practice, in day-to-day practice, when we work in our industry, serendipity happens more often than what we think. It is not a rare occurrence. It happens quite often. Okay. So serendipity and ideas coming out of it still remain one of the biggest sources of idea generation. Second is, of course, literature search. We have to read, we have to go through whatever is available, and based on what is available, we come up with either new ideas or extension of what has been already done. The third source of our ideation, of our thought process, is based on market intelligence. What I mean by market intelligence is that we go and we are constantly in touch with doctors. At the same time, we are in touch with fellow pharmacists. And of course, we are in touch with the patients also, because we keep on conducting various clinical trials. So when we talk to doctors specifically, which are referred to as key opinion leaders, KOLs, we come to know about what we call in the industry as unmet medical need. So when a doctor tells us that, just as an example, I am citing that there is a specific eye drop, and that eye drop actually is a good eye drop, they get good results from the patients. However, the patients complain that the eye drops, when put in the eyes, they are very irritating to the eyes. A lot of burning happens. So when the doctor gives us this kind of a feedback that a specific eye drop has a tendency to cause burning in the eyes, then we start working on it. And our task is to keep the therapeutic benefits of that eye drop. However, the burning should be either completely lessened, or it should be minimized, or if required, it can be prevented altogether. So these kind of things are what are called as market intelligence. Okay. The fourth one which I have listed here is the eureka moment. Eureka moment means that sometimes we are doing something, we are thinking something, and suddenly one thought comes to us, and at that point of time we know that that's what is going to click. So that's what we call as the eureka moment. So this slide is on the step number 1 of new product development, which is called ideation. Can I go forward?
Yes, sir.
Okay. Now, once the idea has come, then the step number 2, which I have listed, is called screening. Okay. Once we know that we have to look for a molecule or a drug which can address a certain unmet medical need, then we start screening those molecules or those products on paper. Okay? It could be a new molecule, or it could be a new product, or it could be a modification to an existing product. That also. What I mean by molecule is a completely new chemical entity. It has never been thought about in the world before. Okay? And first time somebody is synthesizing that chemical entity, it is called an NCE, new chemical entity. A new product is something where a NCE is existing, but a new product or a formulation is being developed for it. Okay. For example, today we know that turmeric exists as a molecule with therapeutic benefits. But tomorrow, if I come up with an injection of turmeric, then it will be called as a new product. So that's an example of a new product. Giving you an example of a modified product, I will probably take you back to Vicks. So Vicks ointment, we have been using for ages and ages, years and years, we have been using Vicks ointment. But many years back, somebody thought about a Vicks inhaler. Now that Vicks inhaler is a modification of an existing pharmaceutical product. A new product or a modified product. This is what I mean by it. Now, once we come up with this thought, and once we have screened a certain ideas to some logical endpoint, then we have to test it whether our idea is workable or not workable. Now, the first point where we test our new ideas is going to the key opinion leaders in the pharma industry, be it doctors or be it pharmaceutical fellow pharmacists. The second thing where we go to is on the regulators. By regulators, I mean the regulatory authorities of the respective countries, whether it is CDSCO of India or whether it is the US FDA of USA. So we go to the regulators, and there is a specific mechanism. We have to send a communication to them. It's called a controlled correspondence. It's called controlled correspondence because it has to be kept confidential between the regulator and the sponsor. So we reach out to these regulators, share with them our thought process, and we wait for their feedback, and we wait for their concurrence or not. The third point is the business analyst. Now, why the business angle comes is because as people in academics are usually very well aware of that, lot of ideas get generated. They make perfect scientific sense. They make perfect technical sense. It's a very brilliant idea. It can be translated also into something which is feasible and something which is very concrete. But just because something has been developed and something is good does not mean that it is going to sell also. We know so many people who have excellent ideas, but whose companies fail. They fail to market the product. Because marketing something is a completely different skill set. So once we have an idea and we know that that idea is good, we always go to the business analyst and ask them whether they will be able to sell this product or not. Okay? So many times a product is good, but it does not sell. And so many times we know that a product is not so good, but still it sells like hotcakes. So that's what we do by talking to the business analyst. The last point which I put here is the scientific modeling. Okay? The scientific modeling is based on various statistical tools, and a lot of them are available. Based on that, we project our idea. We try to find out what will happen if we take it through. For example, if I am thinking about making a tablet product, then I will put through various scientific models and I will decipher whether making a tablet per se would be a good idea or not. So once that science-based model is built, then it gives us a lot of confidence to work forward and take the next step towards the new product development process. Coming to the third point, which is the modified product. As I gave you the example of a Vicks inhaler, this is out of the three things: the new molecule, the new product, and the modified product. Out of these three things, modification of an existing product is by far the most simplest of putting a new product into the market. This reason is very simple. When we talk about a product consists of active pharmaceutical ingredient, we call it API, and along with the API, we are supposed to add a lot of excipients. And once the API is mixed with the excipients, which we call as non-functional excipients, non-therapeutic excipients, non-therapeutic raw materials, that is what makes up a product. Okay, so the API remains constant. Once we have, let's say we have a paracetamol tablet, and tomorrow instead of a paracetamol tablet, I want to make a paracetamol ointment. Just as an example, I am citing it. So paracetamol is known, all its safety, toxicity, efficacy, everything is known. I just have to make an ointment out of a tablet product. Hence, this form of new product generation is by far the most simplest, because toxicity and efficacy are known. Going to the step number 3, it is called concept proofing. Until now, in the first 2 steps, everything was on paper, right? There were no experiments conducted, there was no trials conducted, nothing was done. Everything was on paper. So at this stage, what we do is concept proofing. We collate everything that we have done in the first 2 stages, and we make a concept note. The concept note consists of various technical aspects, scientific aspects, business aspects, regulatory aspects, and then at this stage, a fifth one gets added, that is the statutory aspects. What I mean by statutory aspects is, there could be certain products which are banned in certain countries. There could be certain molecules which may not be suitable for certain populations. For example, the Caucasian population, or the Indian population, or the Japanese population. There have been many instances where, I'm sure you people are aware that Japanese are very tiny people. Okay, they are very small people. Their height is less, their body build is very slender. So what works for a 6 feet or 6.5 feet tall Caucasian white American does not work for a tiny Japanese who will be like 5 feet 2 inch tall. Okay, so we take into account these kind of statutory aspects which vary from country to country. Considering all these things, we prepare a concept note. Next comes the intellectual property. We evaluate whether the idea that we have generated and which we have screened, and now which is appearing as a paper concept note, how to protect this idea so that tomorrow somebody else does not copy it and he does not start marketing it and making money out of it. This is where the intellectual property part comes into play. And the basic fundamentals of getting a patent for anything and everything is, it should be novel. So nobody should have thought about it before. It should be a new thing. It should be a novel thing. Second, it should be useful. If there is an idea which is a completely different, a completely novel idea, but if it is not of any use, then we will not get a patent on it. Okay? So these couple of very basic fundamental requirements are there. And once we are able to have those requirements and the clarity on those requirements, then we file for intellectual property protection on the idea which we have generated. For filing a patent, we have to first file a provisional application, which can be a very, very basic and a very, very minor thing, not required to go through a lot of exercises for it, a lot of experiments need not be done for it. Okay? So once we have protected our idea with an intellectual property, here I think I would like to make one more point. Usually everybody thinks that if you have a good idea, a novel idea which is useful, and you want to protect it, then the only way to protect it is to file a patent for it. This is actually not true. Even if you publish it, it gives you equal amount of protection from getting stolen. Because filing a patent, getting it granted, maintaining a patent costs a lot of time and a lot of money also, which sometimes in academia people are unable to afford to. But they think that they have to patent it, only that is the right way to do it. And in doing that, they lose many, many months of time. My suggestion is that that whenever we get some new idea which is useful, please go ahead and publish it somewhere in a reputed journal, which is a peer-reviewed journal. That publication will give you much more assurance that, and then later on you can follow it up and complete the protection by filing a patent. But do not please wait for a patent to file. A publication is equally important. Okay. Going to the next point. After this, we go to the laboratory. Once this paper concept is done, it is then we step into the laboratory and we start testing the idea by doing some preliminary experiments. We generate some data and then we start analyzing that data. I just want some feedback. Do you people want me to get into more details, or you want me to make it more simpler, or is it okay? How is it?
I think it is good. You can continue with this.
Okay, fine. Thank you. Then I'll go to the next step. The next step is when the actual laboratory work starts and when the new product starts getting developed. Okay. So whenever we start developing a product, first we have to design the product. It's nothing different from how we construct our house. First, we think about something that I want a 2-bedroom house or a 4-bedroom house, whether I want a parking or I don't want a parking. All that is ideation. Then after that, we sit down on a piece of paper and we prepare a drawing from it. And after that, then we start thinking about how to do the interior, what all I will do, where I will keep the bed, where I want to put the kitchen and all that. So that's exactly what I mean by designing a product. And let me also tell you that a product consists of two things. Product consists of a formulation and the pack into which that formulation is going to be packaged. Without pack, the formulation is useless, and without formulation, obviously the pack is useless. So a product consists of formulation and its relevant packaging. So we first design whether I'm going to make a tablet or whether I'm going to make an injection. If I decide that the tablet is the right dosage form for it and I should be making a tablet, then I have to decide whether it will be a round tablet or an oval tablet, whether it will be coated tablet or uncoated tablet, whether it will be dispersible or normal or sustained release or modified release. Okay, this is what we call as a formulation design. Side by side, we also start thinking about the pack design, whether it will go into a glass bottle or a plastic bottle, or it will go into a aluminum foil strip pack, or it will go into a simple blister pack. Whether the blister pack needs to be protected from light, or can it be a clear transparent blister pack. Okay. And most of the times, the pack plays a very important role in the final success of the product. It is just like the king and the kingmaker. So formulation is obviously the king, but packaging is the kingmaker. Many a times, a good formulation fails if the package is not designed properly. Okay? So I always call packing as the kingmaker. Once we have designed and once we know basic things that this is what we are going to do, then we start procuring the raw materials, the required packing materials, and of course the active pharmaceutical ingredient. Okay? QTPP, Quality Target Product Profile. That's what is meant by QTPP. Whenever we design any product, it is supposed to perform, it is supposed to deliver something. So if we want to design, let us say as an example, a tablet for children, for infants. So we want to design a chewable tablet. Now, obviously, there need to be some requirements with which a child will want from that tablet. For example, it should not be bitter in taste. Not only it should not be bitter in taste, it should be good in taste. Second could be, it should have some nice flavor, which children will like, chocolate flavor or bubblegum flavor or orange or pineapple or something like that. Then it should not be very hard. It should be readily chewable for a child so that he can chew it. All these things are example of target product profile. So this is what I want my product to be. I want my product to be appealing. I want my product to have a good taste. I want my product to have a good flavor. Okay, these are called quality target product profiles. So we decide what kind of product we want. Okay. Quality brings in one more thing, not only the physical part of it, not only the taste, smell, feel, all that, it also includes all the analytical part of it. The assay has to be done, the product has to be stable, it should not degrade when it goes out into the marketplace, it should be stable to heat, it should be stable to moisture, all those factors also come up. Now, when all these factors come up, that is where the role of analytical comes in. That even if someone has made a nice flavored chewable tablet of paracetamol, how to test paracetamol in it, how to see whether it is releasing that paracetamol when it goes into the stomach or not. All these form part of analytical development. So formulation development is one aspect, analytical development is the second aspect, and packaging development is the third aspect. Once the formulator makes a tablet, he gives it to his analytical colleagues. They test it, make sure that the right quality and the right testing parameters are built in and they are met. And simultaneously, the packaging fellow also keeps on doing his job and he tries to design a good package for the product. Okay. Once the formulation is made, as I was just explaining, we have to test it whether it works or not. Obviously, the first set of tests is done in the laboratory, which we call as in vitro tests. Once we are convinced that the in vitro tests are satisfactory and are meeting the requirements, then we go to the in vivo test. Now, in vivo tests can be done either in an animal model or they can be done in a human model. Depending on what is the product being developed, how complex it is, okay, what is its pharmacological activity, depending on all those factors, we decide first whether it should be done in animals or it should be done in humans. However, even if we do the first set of in vivo tests in animals, final has to be done in human population. Once this is successful, then we validate. We validate means the simple definition of word validation is that you have to prove that whatever you have done is reproducible, and it again and again, if you do it, it results in similar kind of product, it results in similar kind of performance. That is what we mean by validation. Typically, if one thing is done 3 times and all 3 times the results are similar, all 3 times the performance is similar, then the process is considered to be validated. It still has to be validated further, but the initial requirement is that you have to demonstrate it 3 times. A part of validation, once it is done at a laboratory scale, then comes the next step, which is scaling up the product, which means we are now getting prepared for commercial production of the product. Now, typically, a laboratory scale development happens in the tune of very small batch sizes, 5,000 tablets, 10,000 tablets. Whereas commercial products goes into lakhs of tablets, 5 lakhs, 10 lakhs, 20 lakhs tablets. Somewhere between 5,000 and 5 lakhs, we try to strike a balance, which is what is called scale-up batches. Okay. Once the scale-up batches are successful, along with the scale-up batches, we have to develop the process also, because until now we were only focused on developing the product. We were not at all looking at the process. So the process is of course done while the laboratory scale batches are being made, but that process has to be challenged, that process parameter has to be optimized, and it has to be frozen in such a manner that once the product gets approved, then each batch after batch, when it is produced in the commercial environment, every single tablet that is produced should be of an equivalent nature. So if you are producing a batch of, let's say, 10 lakh tablets, each single tablet has to have the same properties, the same results of all the other tablets, because each patient will be taking only one odd tablet. So that's the biggest challenge when we come to a new product and a commercialization of a new product. Each tablet has to represent all the 10 lakh tablets which have been manufactured for that batch. Okay. Can I go forward, or any questions?
So I think you can move forward. If there are any questions, we'll take up at the end.
Okay, fine. Please let me know how much time do I have?
Sir, take your time.
Continue, sir, no problem.
Okay. No, I will try to stick to the time also as much as possible. But you let me know, huh? If you want anything specific, I can talk about it in more detail also.
10-20 minutes, no problem, sir.
Fair enough. So now I'm going to step number 5. Okay. Until now we have done everything. We have developed the product in laboratory. We have scaled it up. We have optimized our process. We have carried out the requisite amount of testing. Once everything is done, we compile all these things together into a dossier. Okay. That dossier typically runs into at least 10,000 to 20,000 pages worth of data and pages worth of information. So such a huge amount of information is generated. And once this dossier is compiled, then we submit this dossier to the requisite regulatory authorities, whether it is US FDA or whether it is the India DCGI. Along with this, we have to look at the statutory approvals also. When we start a product, we need some approvals. Before we start manufacturing a product, we again have to go to the government and take similar approvals. There are sometimes we have to take some other special approvals also. For example, if alcohol is being used in the product, then we need a license for that. If the product is of a narcotic nature, then we have to go to the Narcotics Bureau in Gwalior and get their approval also. Okay, so we need to ensure that all the statutory approvals are in place. We need to ensure that the regulatory approvals are also in place. Now, once we submit the dossier to the regulatory authorities, they review all the information that we have submitted in the dossier. And typically for regulated markets like US and Europe, this review process can take as much as 2 years to 4 years of time frame. They keep on coming back to us with questions. We keep on answering their questions and satisfying their queries. Okay, it can happen as early as 1 year. It can happen as late as 4 to 5 years. Typical timeline for a regulatory approval is not less than 2 years. 2 to 2.5 years is a typical timeline. Then, before we conduct any studies or any in vivo studies, final studies in human, we have to take the approval of the medical and ethics committee also. Okay. Once all this is done, and once our dossier has been reviewed, then only it gets its approval. Okay. Then the next step comes is of commercialization. Now, when we start commercializing a product, we look at what is going to be its market demand. Okay. And based on the market demand, we have to decide our batch size. For some products, like for example, painkillers, anti-diabetic, the demand is huge. But for some products, say for example, for some products like anti-cancer products, those kind of products, the demands are very, very less. So depending on what is the demand, we design the batch size for our product. Then the last bit which is remaining is the artwork. We call it the artwork. You know, the label design, what we should write on the label, what should be the color which should be put on the label, because there are some scientific requirements on the label and there are some marketing requirements. So those parts are called as the artwork. Then the next thing comes is how to price the product. Now, this is where the entire pharmaceutical industry always has to hear from the public that, you know, we are just into the business of making money. We are not bothered about poor people and all that. However, very few people realize the cost and time which goes into developing a new product. I'll just give you one example to help you understand. Typically, for a simple product, we have to do a biostudy in human beings. A typical biostudy costs anywhere around 1 to 1.5 CR for one product. If we have to do some clinical study or something more difficult, then the cost may go to even 10 CR or 15 CR. Plus all the time which has gone into the development of the product. Okay? All that money has to be recovered by the pharmaceutical company who is developing the product. Taking into account these factors, plus whether there is any competition, what is the cost of API, what is the cost of raw materials, all that is taken together and the pricing is decided. Okay. Then comes the sales and marketing. In sales and marketing, sometimes the company decides to market the product alone. They decide to sell the product alone. Sometimes what they do is, after developing the product, they sell that product to some big multinational or somebody, because a small company may not have that much of sales force and they may not have that much of marketing experience. So the marketing strategy, how to market, who should market, what should be the selling strategy, all that is decided before a product is put into commercialization. Going to the next step, that's of post-marketing. Okay, the job does not end after marketing the product has started. In fact, most difficult part starts now, because once a product is put into the market, the product faces lot of scenario and lot of things which it has never faced before. Because until now, all the trials, all the experiments, they have been done in a laboratory or in a manufacturing setup where temperature is controlled, humidity is controlled, all the machines are validated, they are qualified, qualified people, competent people are making them. So all is a very ideal situation. But when a product goes into the market, it has to undergo, the first challenge is the transportation challenge. Okay? Suppose the products get shipped to Russia or Canada, right? It will encounter sub-zero temperatures. Suppose it gets to Africa or Rajasthan, for example, then it may face temperatures rising up to 50 degrees. When a product is stored by the medical practitioner, the pharmacist, the medical store fellow, many a times we know most of the medical stores in India are open. The sunlight comes in, dust comes in, right? All these factors which are there, they can have an impact on the quality of the product. And this impact results in market complaints. And when a market complaint comes, the complaint is always referred to the manufacturer and he has to deal with those complaints. Okay? Many times people put legal lawsuits on it, that your medicine is not working, or because of your medicine, this kind of adverse event happened, or we got into some side effect, all those kind of things happen. So these kind of challenges come up, which are put either under a medical legal challenge, or it can be simply put across as market complaints. Okay? We have to manage the supply chain and we have to manage the changes in the market. I'll give you one example of changes in the market. Just 1 month back or 2 months back, there was a severe shortage of the medicines which are required for COVID. For example, amphotericin liposomal injection or remdesivir injection. All of you know that remdesivir injection used to sell for 30,000 rupees, 50,000 rupees per injection. Whereas otherwise its real market price is maybe hardly 2,500 or 3,000 rupees. But now suddenly everybody has remdesivir, everybody has azithromycin, all the medicines are available and they are available at the MRP. So when such kind of market changes happen, suddenly the manufacturer of the medicines, they face a lot of stress and they face a lot of losses. The third factor which can many times happen is price crash. Okay? This is not very likely to happen in our country because here our prices are regulated by the government. But in US and Europe, there is a free market. You can sell your product at $100 also, and if you want, you can sell your product at $10 also. The government will not interfere into it. But what happens is when I'm selling my product at $100 and two more companies come up and they start selling at $20, so I'm forced to sell my product also at $20. That's what I mean by price crash. Same thing happens when increased competition comes in. Okay? When there is just one or two players, they make more money. When many players come in, the competition rises. Okay? The next point which I have captured here is the viability of the product. Okay? Now you must have heard that there have been a lot of changes in the COVID management protocol made by the WHO. So the medicine which was into that protocol today is out of that protocol. So the product no longer becomes viable. Okay? Same way, sometimes another thing which happens is with viability of a product is that many times the product is very good, but the market is unable to sustain that product. For example, I'll tell you, transdermal patches. Okay? I mean, the research on transdermal patches is going on even before I was in college. More than 50 years back, transdermal patches are being done. But even today, if you see, the number of products of transdermal are extremely limited. Because if I make a transdermal patch, let us say for motion sickness, okay, people will say that, ondansetron tablet is available for whatever, 2 rupees one tablet. So why I should buy a patch which will cost me 200 rupees? So they just don't buy that product. So that is why even if a product is good, it does not remain viable into the marketplace. So all these scenarios have to be taken into account, which is what ultimately decides what we call as a life cycle of a product. So many a times there are products which keep on selling for years and years and years. Whereas there are some products which are just unable to sell. They just crash, they just die their likely death. The last one is, I think, the step number 8, which is the life cycle management. So when I told you about this, a product dying its likely death, that is what is called the life of a product. And life cycle management means how much I can extend the life of my product, just like a human life. It is just if you make some innovation to it, if you do some continual improvement in your product, if you block your competitors, there are many ways in which you can do life cycle management of a drug. A perfect example of a life cycle management of a drug and extending its life is I will again come back to, let's say, Iodex. Iodex used to be available as a jar of ointment. Then came the spray of Iodex. Okay? Now, after a few years, somebody will think of something else of Iodex. So when these kind of innovations are done in a product, the life cycle of the product gets extended. Same way, it's not that the innovations can be done only in the product. Innovations are done even in marketing. Okay? The marketing companies, they keep on evolving new ways. They keep on evolving new strategies for marketing a product so that the life of a product gets extended. The last one, of course, is to keep on improving on your product itself. There are some complaints coming for the product, you resolve those complaints, people will keep on buying your product. But if you do not resolve the complaints of the consumers, of the patients, and of the doctors, then obviously that product will not be successful. So all these strategies companies use to extend and manage the life cycle of a product. Okay. So finally, the summary and the conclusion. Okay. A new product development is an extremely complex process. There are a lot of moving parts into it. It involves a lot of money. It involves a lot of time. It involves expertise of various functions of research and development. Testing and validating each step of new product development is essential so that finally the product can be commercialized batch after batch in a successful and a reproducible manner. In the new product development process, the stakes are very high. The losses are very high. If a product succeeds, then it is like a gold mine for the pharmaceutical industry. But at the same time, if a product fails, it can incur losses for the company, which can even take the profits of all the successful products. So the stakes are very, very high. The risks are very, very high. Okay. We have to consider the factor of science versus business. Okay. Science may say that this is good, but business may say it is not good. Okay. So that factor is very, very important. Even a good science-based product does not necessarily mean that it will be a business success. Okay. Continuously, we have to keep on reading and keep on updating on the regulatory guidances, particularly from the renowned regulators like the WHO, US FDA, or the International Commission on Harmonization, ICH. The industry-academia collaboration. Now, this is a very hotly debated topic. I have been called and I have spoken in so many symposia and conferences on this topic, industry-academic. The problem about this topic is we all know what is wrong, but we do not want to fix what is wrong. Okay? In my opinion, industry and academia are like two tracks of a railway. They run in parallel, they run together, but they never meet. And they ensure that the train runs on them and the train reaches the destination. That is at least what is required that they both ensure. So industry and academia, in my personal opinion, they can never meet, simply because the thought process is completely different. The culture is completely different. But it is okay to be different as long as the end objective is achieved. So I always give this example that the best way to meet those two railway tracks, the people who can make those two railway tracks to meet is the birth of the railways, and those births I consider as the students, because the students are there in academics, and then once their academics is completed, then the same students come and join the industry also. Okay? So this topic of industry and academic collaboration, they need not meet, they need not agree, but if each one does its own job, then the ultimate aim of the train going forward can be achieved. Okay? Similar situation is with the pharmacist and doctor collaboration. Okay? The doctor has to do his job, the pharmacist has to do his job. But what we keep on fighting with each other is, we as pharmacists always feel that we are underlined, we are sidelined, we do not get the requisite importance, the doctor gets a lot of importance. So we always keep on complaining about this part. Okay? Same way, the doctors are always in a superiority mode. They feel that they are the ones who are God, and they are the ones who are treating the patients. So this collaboration of pharmacist and doctor, to the extent that it should happen, is not happening. As I gave you the example at the start of my talk, the best teacher for us in the industry is actually the doctor. If the doctor tells us what is good and what is bad, then we as pharmacists can do a much, much better job. So these four pillars, industry, academics, and the pharmacy and the medical profession, if these four pillars are able to work together, then I think the end objective of a proper and a good health care for the human population can be achieved. All right, that's what I have from a presentation perspective. And now I'm open to questions or any other point that you want me to talk about.
Well, Dr. Jain, thank you very much for a nice presentation. And in fact, if I look at it from the student's perspective and also from industry perspective, you have summarized the things very clearly and beautifully. If I have to quickly
now session is open for discussion. If any question.
Yeah, I think your voice is breaking. I could not hear you.
Voice is break हो रही है डॉक्टर साहब. Dr. Sunil ji.
Yes, sir.
Now okay, voice break हो रही थी, now okay. So if any question on YouTube or any person, those who are direct connect with the Google Meets, they all are welcome. If you want to any queries, any question. Sir, from my side, number of time we are discussing in the academia for the industry academia interaction, industry academia partnership. But I feel that when I approach to the industry, then I feel a number of the difficulty and most probably the, we can say, attitude or orientation or like that. The industry feel that academic people's what they are performing in the institute or publication. So somewhere is the big gap, and they are not believe on the data of the academia like that. But I feel that. So as you know that ISF College of Pharmacy, we have the lot of facilities, and I also from in this platform I want to invite some of the pharmaceutical companies so that we can do the some of the collaborative work, because we have a lot of infrastructure as well as human resource. So how we can approach to the industry so that we can reach and we can give our availability of the facilities, equipment, human resource, and how we can contribute for the industry. So how we can Karajgi sir.
Yeah. So I think your question is a valid question, and I will tell you why this problem happens where you feel that you are not getting enough support or that attitude mentioned, because usually in industry the right person. If you do not approach the right person, then you will keep on going round and round and you will end up nowhere. So it is very important that such things be done more on a personal level first, and once you get the right contact in that particular industry, then your things will become easy. Okay? Because let's say as an example in my company itself. Now in my company, whatever, there are 5,000 employees who are working with us. There is an HR department, there is a business development department, a lot of different people are there. It is very important that you touch base with the right person. What happens is the person many a times is the decision maker on such matters. Okay, so if you approach the right person, then the right thing can happen. Secondly, you come into your topic that industry feels that the data generated by academics is not reliable or whatever. This is a very common problem which we face. Okay, and the best way to overcome this problem is to invite the industry to do an audit. So if you tell that I have this idea, you please come over and you check whatever is required, then there will not be any problem like this. Because you people in the academia have time to think, you have good brains, you can think. The problem with us in the industry is we don't have time to think. So if you do the thinking part, and if you do the ideation part and the screening part, the remaining steps will be done by us. That is how would be the ideal mode of an industry academia collaboration. So first two, three steps you do, remaining 5-6 steps I will do.
So thank you very much, sir, and I will try my best. Sir, this is for just I want to give the information. Sir, this institute received the 10 lakhs grant. The one of the project, this is the industry academia partnership, and sir, we are also running of the business incubation center approved by the MSME. So step 2, how we can connect the industry. So your lectures and your advice, it is the very helpful to the institute and also to me. And you have just mentioned but one key right person. So this is the again question in my mind, because most of the academic person, they are connect with the HR. So please, if possible, if you can give some of the clue like chairman, president, or some of the another responsible person, who this department, or you can say like a post wise or by the department wise, they are looking this types of the activity. When we connect to the any industry, they feel that एक हिंदी में कहूं कि इंडस्ट्री को ये लगता है कि इंस्टीट्यूट सिर्फ मांगने आया है.
Correct.
Either they can be asked to me some of the sample of the drug sample, or either they can be asked some of the chemicals, some of the polymers, or either only for the visit of the institute. So these are the also activity. This is also requirement of the academia. But other than this, we have also some of the good thing like a collaborative activity. So how we can approach it, but if you can give the some name of is very helpful to me. Or most probably we are the connect with the HR. We submit the application, detailed proposal, profile of the institute, but I never received the response from any industry, sir.
Yeah. वही चीज हो गई जो मैंने बोला था. You have to reach the right person, and HR is not the right department. And that time you people don't get the answers. Okay. Typically in any industry, if you want to be heard, then you should approach the head of R&D. Head of R&D is the person in industry who will understand.
हो रही है, sir, please.
अच्छा, एक minute. Is it better now?
Yeah. Now this is better.
Okay. So I am saying that instead of HR, it is always good to approach the R&D person, the senior most person in R&D, because he understands academia. What I think you should do. And on a case-to-case basis, you should use your contacts wherever possible.
So thank you very much, sir. Now, any question, Dr. Sunil ji?
There are no questions from YouTube also, sir. So
okay, so thank you very much. So now over to Dr. Sunil ji.
Jen sir, I was just summarizing the presentation, and it was quite a beautiful summary of new product research, new product development, as well as the new product launch. And, we have started very nicely with the
presentation को close कर दीजिए सर, जैन जी.
Sorry.
आप अपना presentation sir.
हाँ.
Stop the sharing, sir.
Okay.
Sir, thank you very much.
So very nicely you have depicted about the ideation, and without specifically giving and another during the screening phase, as you indicated, like different formats of Wix. I was just wondering if we can make use of the lipstick format in some form. Then coming to the proof of concept, where R&D and marketing people usually they work very closely, and I think this is the phase where the new product research ends, and then we move on to the commercial side of it, where the new product designing starts. And again, one thing which I found was implied is force degradation studies along with the method development. And then coming over to statutory approvals and technology transfer, again, it was very nicely implied in your communication. Then during the commercialization stage, my experiences with marketing and sales people are slightly different. And because as R&D person, we work very closely with marketing. And on the day of launch, when it is more with the sales team, marketing people have different opinions. So R&D has to sometimes suffer on those accounts.
Correct.
And post-marketing thing, the supply chain steps to plan the web experimental batches, considering when the advertising and other things are to start, and 90 days supply chain things are required. And finally, the life cycle management, with where you have indicated about the market innovation, the problems. It was very nicely captured, and especially the end I liked the 4 pillars: the academics, the industry, and
the doctor and the pharmacist,
the regulators, and the other part of it. And thank you very much, sir. I have heard your name many times, but for the first time I got to see you. Thank you. Thanks a lot on behalf of ISF.
Thank you. Thank you. And if anyone wants to reach out to me on a personal level, you are most welcome.
So thank you very much, sir. And this is my privilege. And as I know that when I was the student of B.Pharm in 1989, so Dr. Jayant Karajgi ji is my senior as well as my teacher also. सच बताऊं सर, जैसा आपने कहा, you can connect to me number of time I have tried to connect to you, but some of the internal factor, or you can say fear, that because this types of the personality or the position, they have very short of time or they have the no time. I interact with the I just connect with the Singh sir. He told no problem, so I just I just called them to give my phone number and email. Then I connect to Jayant Karajgi sir. So thank you very much, sir, for accepting our invitation and your motivational lecture. And this is the very, very highly scientific, direct related to the how we can develop of the new product, commercialization, market problem, and price crash, and so many things you have included very simple and specific manner. So I hope that any person follow the system and he or she they can be achieve the goal. And I also request from institution side, I will connect with you, and I also approach your company with through you, so that how we can collaborate ISF with your Silpa healthcare system, so that we can do some of the good new things. And from my side, I will provide all types of the things which you feel that you have some of the doubt related to the academia. Now the academic system, they are grow in a scientific manner as also. I am not to ensure the all institute. Why I ensure from my side, if we can do any commitment, I will do it. Sir, I want to include over here. This is, as you know, that Sri Ravi Shankar ji, and is one of the pharmaceutical company Ayurveda. He connect to me and he did the MO with me, and he also performing some of the toxicity study with this institution. And Professor Gupta ji, he is a president AIIMS-Bhopal and Jammu. He is also connected with institute as a advisor and chairman of the Research Advisory Council. So he is the regularly giving number of the ideas like that. And yesterday, Dr. Anil Kharyaji, he interacted with the YK Gupta ji, and he discussed number of the things related to Jain and some of the difficulty related to the government for the new product like that. He also give the number of the ideas, so that you can develop some of the product which are related to the protection or for the treatment of the COVID like that. So I hope that in coming future we can do some of the good job in the field of the research and development of the dosage form, and somewhere we can also did. Here you have mentioned what very important, what is the packaging of the product. So itself there is a lot of work even in the packaging. So thank you very much from core of my heart and ISF family, sir. And I hope that very soon we can meet we invite you, so that we can meet over here and also do some of the good work, sir. So thank you very much, sir, for accepting our invitation, and Grace Deepak.
Thank you. Thank you everybody for calling me and hearing me. Bye.
So dear friends, the announcement to all of you, the valedictory function at 4 p.m. and our chief guest, Colonel V. Venkat, Director FDC AICT New Delhi. During the valedictory function, we announced all the results of the poster session, and our experts of the poster session, they have given the result to our organizing team. And each section we have given the 3 awards, best poster award. So total section is the 3, but in the pharmaceutics and pharmacology, there is the number of poster, or you can say number of poster available. That is, our team has decided to give the 4 number in pharmaceutics and pharmacology, and also 4 best poster award in the category of the nanotechnology, because 2 position is the tie. So I request to all participants, you can connect with the YouTube, and our team will send some of the message or some link. You can join also through the Google Meet. So thank you very much, and thanks to all.